Okay, are we ready?
I think so, yeah. So we can get the ball rolling.
All right, excellent. Okay, welcome.
Ken just joined. Okay, I'm going to mute myself and let you do your stuff.
Okay. Hello and welcome to the Carboncopies Foundation 2019 Summer Workshop event, Updating the Roadmap to Whole Brain Emulation Part 2, Where We Go From Here. This event is produced entirely online with the help of our generous patrons and donors, as well as our team of talented volunteers. I am Michael Ulrich, the Carboncopies Foundation Event Director, and I will be your moderator for today. This event is the second installment of our series for updating the 2008 Oxford University Roadmap. For this event, we'll be taking the advancements we have discussed and exploring the next challenges facing the development for whole brain emulation since our previous events. We aim to set up the next course of action, define the next problems, and set goals for solution to those problems. Our agenda for today will be as follows. To start off, we'll have an introduction prepared by the Carboncopies Foundation Chairman, Dr. Randal Koene. At approximately 8:15 a.m. Pacific Time, we will play an interview recording with Professor Konrad Kording. Professor Kording heads up K-Lab at the University of Pennsylvania. He is known for his work on the intersections between computation and biology, using machine learning to explore neuroscience, brain mapping, and neural data. Professor Kording also likes to take a step back and give a skeptical perspective on neuroscience, as it is practiced today, as evidenced in some of his recently co-authored papers. What does it mean to understand a neural network? Reverse engineering neural networks from many partial recordings, and could a neuroscientist understand a microprocessor? Then, at approximately 9:25 a.m. Pacific Time, we will play an interview recording with Professor Dong Song at the University of Southern California. Professor Song has worked with the team of Professor Ted Berger and collaborators to develop a medical neuroprosthesis for subregions of the human hippocampus. This work has been tested with successful results in studies with patent volunteers and is supported by DARPA. Professor Song specializes in the analysis of neural signals and in machine learning and statistical methods. Following the interview, we will have our panel discussion at about 10 a.m. Pacific Time, followed by our panel Q&A session. Then at approximately 11:30 a.m. Pacific Time, we will have our final panel discussion, followed by the final Q&A session. Dr. Koene will give closing remarks at about 1 p.m. U.S. Pacific Time. Our workshop is participatory and is open to all of our international attendees and viewers. You may ask questions, and when it is your turn, you can opt to engage directly with our panel, or you can leave it to the moderator to repeat your question aloud. You can write questions directly into the chat that is enabled for you on our YouTube live stream. One of our excellent staff members is monitoring the chat and will alert me to interesting and topical questions, which I will read to the panel. More complicated or multi-part questions where it is important to establish that the question is properly understood and answered are best asked by engaging directly with the panelists. We make this possible through our call-in number. The number and PIN are shown at the bottom of the screen right now. They are also written in the livestream description and on the livestream.carboncopies.org webpage. They are 786-540-5631 with PIN number 180-618-986. If you are calling from outside the U.S., you may also use the web call-in option at call.carboncopies.org to avoid telephone charges. This process is similar to calling to a radio show. Your call will be monitored, and a respectful exchange is expected. When your question would benefit from direct interaction, I will invite you to call in if you can. If you want to add a note to your question stating that you call in or that you cannot, that can be very helpful for our moderator. Our many thanks go out to our patrons and donors, to our volunteers, and to the many experts to make time in their busy schedules to participate in our workshops. We are a 501c3 nonprofit organization operating with the sole mission of advancing research in whole brain emulation. We appreciate the many contributions that allowed us to organize events such as these. If you would like to become a monthly donor or to make a one-time donation, you can visit carboncopies.org/donate and donate by PayPal, credit card, bank transfer, or cryptocurrency. And to start off with the introduction, Dr. Randal Koene, the Carboncopies Foundation founder and chairman, has prepared an introduction for this event, and I will play that introduction video for you now.
Where we go from here. Whole brain emulation is the technical term for a process by which the very specific functions of a brain can be recreated so that a mind with particular characteristics, memories, and experiences is produced. It is the process for mind uploading. In all likelihood, whole brain emulation is physically possible, though very difficult by today's standards. The beginnings of what will be involved to achieve a whole brain emulation are visible today in the efforts of neuroscientists. Indeed, we could say that whole brain emulation should be a natural outcome of successful neuroscientific study, as the domain of neuroscience matures toward a complete understanding and an ability to computationally model brain mechanisms. Consequently, my response when asked about the timeline for whole brain emulation is often this: I would be extremely surprised if it was accomplished within 20 years, but I would be equally astonished if it did not happen within 100 years, assuming that infrastructure, economy, and science continue as they have. By historic, evolutionary, and certainly by astronomical standards, 100 years is not even the blink of an eye. In other words, if we assume that humanity continues to thrive and strive, then we can also assume that humanity is now on the very cusp of achieving whole brain emulation and by extension mind uploading. Just a little over 10 years ago, in 2008, the first attempted roadmap towards whole brain emulation was published by the Future of Humanity Institute at Oxford University. I was involved in that publication, which featured the collected and edited output of a group of researchers who convened at Oxford in 2007 for the first workshop on whole brain emulation. The first roadmap was necessarily incomplete at the time. It focused heavily on a subset of the problems of whole brain emulation. It is time to compile the knowledge and the data for the updated version of the roadmap. To that end, on June 16th of this year, the Carboncopies Foundation launched a series of events that are aimed at presenting updated accomplishments and updated understanding to the public in preparation for the new version. In our first event of the series, Professor Cho from Texas A&M gave us a peek at the scientific process for a better understanding of the phenomenon of consciousness by devising models of neural dynamics that can predict aspects of consciousness. Professor Song of USC revisited patient-specific prosthetic neural model building, and advances of memory decoding experiments with the neuromimetic hippocampal prosthesis. Professor Parker, USC, reminded us of the importance to test the functional relevance of characteristics of biological neural signaling. Dr. Decca, of Neurobotics, presented grid cell responses as an example where evolved coding, in this case for space, may enable superior autonomous navigation. She posed this as an example of the interplay between emulation of brain mechanisms and insights for AI. Dr. Hayworth revisited the state of the art in brain preservation, which has reached a point where reliable long-term brain preservation appears to be both possible and practical. Hayworth also emphasized the importance of whole brain emulation proof of concept studies and the overall need to build consensus around the connection between fundamental neuroscience research and logical outcomes with meaningful impact in the world. Prominent neuroscientists must take responsibility for this reality. The panel discussion then turned to the obvious question: where do we go from here? Creating working models from extracted data and validating results according to improved success criteria were foremost on the list. The second event of the series is tentatively scheduled for September 21st. It is titled Updating the Whole Brain Emulation Roadmap, Part 2, Let's Build the Emulation. It asks the question: imagine it is the year 2030 and we can collect all of the brain data that we expect to be able to collect. How do you use that to do whole brain emulation? The event will feature interviews and panel discussions, and we will explicitly ask the participants to take that step, to look beyond the predictable next engineering step and instead at the more significant problems that still need to be solved. So that we may separate philosophy from physics, we will address this for a tiny animal first, before we ask about human mind uploading. In the June 16th event, we already learned that aldehyde-stabilized cryopreservation may be the solution for the long-term storage of brain tissue in a manner that preserves every relevant detail of the structure of the brain's connectome. We look at that and we say brain preservation is scientifically a solved matter. Turning it into a reliable process that can be used for human-sized brains is an engineering problem that will make almost predictable strides in the coming years. We can write about those details, the milestones that are the peaks and valleys in the roadmap, but we don't need to worry much about discovering a path. We also learned about multi-beam and focused ion beam electron microscopy technologies that are multiplying the volume of brain tissue that can be accurately scanned in a reasonable amount of time at resolutions that show the details of the brain's connectome, which our consensus assumptions about whole brain emulation deem necessary. Certainly, there are still some questions about reliably identifying what has been scanned, or about methods to label what will be scanned via chemical or molecular means for such identification. But the capacity increases that are needed to get to the point where imaging the connectome of brain tissue is possible at the scale of a human brain are looking more and more like engineering challenges instead of fundamental scientific questions. Again, we can write a lot about those details, but we're less worried about discovering a feasible path than we might have been 10 years ago. The first roadmap didn't say much about electrophysiology or about collecting electrophysiological recordings from a very large number of sites throughout a living brain. That is a type of data collection that is crucially important to the characterization of brain mechanisms and systems. The road there still looks a little murkier, despite the efforts of the Human Brain Project, the Brain Initiative, the Allen Brain Institute, and many others. Multi-site recording is beginning to take off, but its ultimate potential and the optimal technology path is still a little uncertain. There may be several paths worth taking. Therefore, there is some terrain the roadmap should suggest to explore. Notice that I haven't mentioned any models yet, any emulated brains or emulated pieces of brain. Those models are where it all has to be put together. In previous events, the Carboncopies Foundation has frequently highlighted the efforts by the Berger Lab and its associates to create a neuroprosthesis for regions of the human hippocampus. We did that specifically to highlight this important problem of bringing the structure, the characterized functions, and the desired functional output together in one model, in one emulation. The Hippocampal Prosthesis Project is an attempt to create such models, such emulations of a specific patient's brain function, and then to cast those into medical devices tailored specifically to that patient's needs. The results are preliminary, though promising. The chosen system in regions CA3 and CA1 of the hippocampus is purposely structurally relatively simple. It means that researchers working on that prosthesis were able to focus on model building by combining well-understood structure with characteristics of recorded data. That hasn't been possible in most other parts of the brain where connectome details still need more data and more effort to reconstruct. The retina of the eye is one other relatively simple region where prosthetic modeling has been possible. So this is where we begin to run into a multitude of scientific and applied science questions. There are many possible paths to follow and probably many studies to conduct. We would like the updated roadmap to describe not just the terrain historically covered, or the known engineering paths with predictions for the time from capability A to capability B. Rather, we would like the roadmap to look ahead to our goal. We would like it to include the best ideas for exploration, and so we would like to ask the pioneers to consider the unknown pieces. Dear pioneer, come with us into our virtual landscape. Imagine that you find yourself at the end of the path that has been taken so far, and even a little further, as far as we can comfortably see along the path. So please imagine that it is 2030, and we have reached the point where brains can be preserved virtually forever without any damage to connectome-relevant matter. Please also imagine that scanning of brain tissue at electron microscope scale has reached the point where the details of entire brains can be digitized in reasonable time and at reasonable cost. Perhaps these are Drosophila brains in 2030. That's fine. Let's also assume that labels and other identification methods allow us to automatically process the image data into three-dimensional maps that show the locations of, the spatial dimensions of, and the connections between all of the identified scan content. Imagine, therefore, that we have in 2030, or is it 2040, a process that can convert the preserved tissue of a brain into a reasonably reliable and accurate full connectome, synaptome, etc. tome of that brain. Let's first assume that we want to create a working whole brain emulation of a Drosophila with its 135,000 neurons. Where would you take us next on our exploratory journey to this goal? Keep in mind that the maps created with the data that was acquired are not working models. They are a collection of data without processes to run. They are the image of the sea, not the sea roiling under the influence of gravitation and winds. Perhaps, first it would be a good exercise to know a little bit more about the goal. For our whole Drosophila brain emulation, what are the success criteria that we use to determine if a successful emulation was built? Is there a system process that you can imagine, or systematic process that you can imagine, and which we can then discuss in detail for the updated roadmap, by which all of those brain tissue maps are converted into processes, including the constraints and means of validation that are needed to determine that the resulting processes are not just any processes, but are indeed those that generate a successful whole brain emulation of that specific Drosophila. What are the main difficulties you can imagine? For example, are you at all worried about justified scale separation in a complex system? Are there important alternate routes to consider that you can already see in the distance? That was quite the hike. Awesome. Now, for the next step, let's assume that we want to create a working whole brain emulation to upload your mind. Are there any new success criteria that need to be met? And do those raise any additional difficulties that we should discuss before we embark on that journey? Thank you. We'll be poring over this wonderful travelogue, and then we'll be back with our next set of questions as we try to collect what we need to know for that updated roadmap. Your travels will tell us a lot about the updated state of what we might call the consensus approach to whole brain emulation. Several parts of the approach will have moved from scientific problems to being largely engineering problems that present a clear progress curve toward the required capabilities on a somewhat predictable timeline. Others will still contain basic scientific questions, even though the consensus about underlying assumptions is still strong. For example, that cognitive functions are accomplished by neurons that interact through the exchange of electrochemical signals, and that a separation of scales is possible, whereby some aspects of the biological machinery behind that interaction are not in themselves crucial to the cognitive functions experienced. Finally, there may be some areas where our understanding has changed in important ways over the last decade, with new findings and insights that challenge specific assumptions of the consensus approach. So let's just start with a few things that you've recently been a co-author on to see how they fit in here. I was noticing that you recently published a paper called "What Does It Mean to Understand a Neural Network?" And I was very curious what your take-home message of that paper is.
So for me, the take-home message is that...
So Michael, while you're spooling up that next one, I just want to remind any one of our speakers or panelists who have joined the meet that if you want to see the interviews being shown, you should head to the live stream on YouTube, because that isn't also being streamed back into this meet session.
Like an outstanding neural networks person. And we get talking about what it would mean for us to understand the brain, for us to be happy with the way we understand the brain. Yep. And so we started asking ourselves, what would it mean to understand a neural network? Because like hey, like the brain is like pretty abstract. I have trouble wrapping my head around what it would mean to understand the brain. But in neural network, that's something like that we built. That therefore we can meaningfully talk about what it means for us to understand. Mm-hmm. So we basically said, okay, let's ask ourselves what it means to understand a neural network. Now, do you think we understand neural networks?
This is a very good question, and this used to be a big topic back in, say, 1996, when people were saying things like, oh, you will never convince someone to build an autopilot that lands a plane based on a neural network. You'll be able to demonstrate to them exactly what the formula is that the neural network is using to land the plane.
Right.
Yeah.
No, so it is a very old question, but the interesting thing is that neural networks, at the same time, we totally understand them and we have no clue about them. Let me break down what that means. So, in a way, we totally understand a neural network. No, so if I build a neural network that, say, recognizes objects, it might be 1,000 lines of PyTorch code. Mm-hmm. Those 1,000 lines, I can totally really understand what's going on there. And I can sit you down. I can say, hey, let's talk about this. Let's go through that. And I sit you down and I go through the source code. And like an hour or two later, you'll be like, yeah, I now understand what all the lines in this code do. And in a way, that's understanding. But let's talk about the nature of the lines. What the lines will be, it will say, let's try and be as good at recognizing objects as we can be. Let's calculate what an image is, and if we made a mistake, let's improve our parameters. More specifically, we might be doing stochastic gradient descent, but it basically just says, I take this cost function, which is like maximize how good we're at predicting if something is an image, and minimize that cost function, so like have the fewest number of errors that we could have. And to do so, we calculate the gradients and we take small step into the right direction. And by doing that often enough, we're going to get very good. And in fact, if we implement that code, we take that code, which is these 1,000 lines if you want, and then we give it 1,000,000 images. Some of images might be cats, some images might be dogs, some images might be a photograph of your or mine face. Mm-hmm. And we train it to be good at recognizing such things. And in the end, it does get to be very good. So at some level, we understand it. No, like we built the code that does it. And in a way, we don't understand it at all, because after it has learned, it will have millions, maybe 100 million free parameters that are all set through the learning procedure. Mm-hmm. So it starts with like some random initialization of them, and then it changes the weights, makes some of them bigger, some of them smaller. So in the end, it gets like so many images right. It might be doing it better than you or me. But if I ask you, well, what's the role of weight number 10,369 in there? I could just say, well, it's the weight between neuron like 17 and neuron 519. But I can't give meaning to that. And so the interesting thing is, and like we have this contrast that we fully understand the generator, which is, we can say we meaningfully understand it, which is we can do things with it. If the network makes mistakes or doesn't train well, we can use our knowledge of having built it to make the network better. If the network has some problem, we can fix it. Mm-hmm. And yet, if I ask you, well, look here, you have like millions upon millions of parameters. What do they do? What does any one of them do? Yeah. No one in this world can explain to you what it means. Yep. And that same problem is with the brain. Now, you could say maybe the brain can be understood at that meta level, where you could say, well, what we do is we basically, as part of recognition, we try and become good at like recognizing objects. Mm-hmm. But if we have these simple things that basically just, if you want, that the brain has rules of making itself better based on the experience that it has, what these simple rules produce is something that becomes arbitrarily complicated. So that after it has learned, we might not have any hope at being able to understand it.
Right. Yeah. Okay. And, then someone will say, sure, that's true for your arbitrary neural network. And it sounds a little similar as if you're describing, okay, in nature, we want to understand how an animal catches its prey. And we can say something like, well, we think that, this part of its brain is constantly calculating a trajectory and trying to get ahead of the prey to find where the prey is going to be next, without describing exactly what's happening in every part of that procedure. But at the same time, you don't tend to feel quite satisfied with that. So for instance, when it comes to understanding vision, we don't just say the network is learning to recognize objects and it's using this learning rule. Instead, we go in and we try to find what are the different layers doing, V1, V2, V3, V4. What are they actually focusing on? Oh, look, this is picking up T-junctions or edges or something like that, and movement to the left or movement to the right. And then we still plug that into a network and say, okay, now, using something like this as the input, what can we do with a learning network?
But are we? So here's the problem. So if you have a network that you train to solve a task, you will find neurons that have some tuning. Like you will find maybe somewhere that there's neurons that are more active for the red stimuli that you happen to have than the green stimuli that you have. Mm-hmm. But that assignment that you just said, we have a brain area that does T-junctions in a system that learns. That may never be true. So we might be fooling ourselves believing that there's simplicity at that level. Like we have never found an area that does T-junctions, and we have never found an area that does color. Yeah. We find an area where we find that there's a good number of neurons that have different activity if we show red or green or blue or yellow. But what we have been doing in the past is a fundamental logical mistake. What we did is we showed different colored stimuli where we said, okay, let's go red, let's go green, or look, there's a difference. And then we said, because there's a difference, this brain area solves the problem of color. In the same way as you can say that brain area solves the problem of T-junctions. Sure, if we go to the different brain areas, we will find some areas where T-junctions have more activity than in some other areas. But we impose our human sense of simplicity on that. Mm-hmm. And that sense of simplicity is what's leading the entire field astray. And you can see why we came up with that wrong idea. The reason why we came up with that idea is if you're in a world where you can only record from one neuron at a time, and you assume that neurons don't do something simple that you or me would have words for, but that neurons do something truly complicated, we have no hope of understanding the brain. Therefore, that assumption that every brain area does something simple, if only we discover that we understand the brain, had an enormous pull because it allowed us to basically sell that we can understand the brain based on it. But this notion of simplicity is dying across all corners of neuroscience at the moment. So if we look at individual neurons, it's not that this is a color neuron. No, a large part of the brain cares about movement. Just about every area cares about reward. It's basically what we are finding in a neural network. At just about every layer, there is a lack of simplicity. And in the same way is there a lack of simplicity if we look at the brain and don't wear rose-colored glasses. Mm-hmm. Okay. And this mistake of the belief in simplicity of brains was extremely at the forefront of neuroscience. It's been guiding it. And we found lots of we found areas that are specialized for certain things. We found areas where neural responses look somewhat similar to one another. So it's been a success story on the one hand, but at the same time, we are fooling ourselves if we believe that we can get from there to what we would feel is a meaningful understanding of the overall system.
Mm-hmm. Yeah, if someone were to come into this conversation and not know all the background, then it might sound as if you're railing generally against reductionism. That's not the case in this, I think, because, for instance, even if you're not going to say this these neurons in this area are all trained to recognize green or something like that. You can still say that these neurons, or a certain subset of those neurons, learn to recognize green because of the way they were subjected to stimuli and what the learning that they underwent. You know, the stages of learning, say, from when you appear in the world as an infant to when you can recognize letters on a chalkboard.
Yeah, so let's talk about a success story in that area. Let's say we talk about early visual cortex. You can say, we can look at the outside world and we find that it is largely dominated by little edges. You know, like, look around your office at the moment. There will be lots of edges all over the place. And now you can say the representations that the brain has at its early layers look a little like they look for edges. And if you look at the representations that you have early on in a neural network, well, they also look like they're looking for edges. More precisely, they both look a little bit like GABA waves. Not perfectly. There's also color and motion and all kinds of other things. But like first order, they look like that. And if we then assume that it's a good strategy to represent what we have in the world as a sparse combination of things, we can say a meaningful way of describing the world is kind of an addition of lots of edges. And work like the work of Bruno Olshausen has shown that we can understand a good aspect, a part of the properties of, say, primary visual cortex by assuming that neurons basically locally explore GABA wavelets. Mm-hmm. But that is a beautiful story. But it is a story that describes a very small part of the variance. And that is where we run into trouble with the traditional reductionist framework that we have in neuroscience. And just as an aside, for whole brain emulation, we wouldn't need that. No, like, we don't need simplicity.
True. Okay, we could debate that because it depends on your implementation strategy. For instance, it depends on, say, if the manner in which you would be applying all of the details for a reconstruction of your brain, if that was reliable enough, or if you need to correct errors by applying on top of that some templates of typical architecture, typical function, right?
But let me push back on that. I think that that is the wrong way of pushing. So you could say, if I missed some data, I could, in one scenario, say, well, neurons in that area happen to usually look like edges. But alternatively, I could say, this neural network, I know certain weights, I know certain aspects about it, I'm uncertain about others. Let's change the network until it gets to be good at solving real-world tasks. This is a much softer way of biasing towards the right direction, but if you want, it is a much more meaningful one because you're actually fixing the kind of things that are also useful otherwise. Yeah. So I'm saying pushing towards a simplicity that people project in their own mind is the wrong strategy to that, because that simplicity is an illusion. Whereas pushing towards stuff that actually works is going to be universally useful.
Mm-hmm. Yeah, I tend to agree with that. I think that's the difference, for instance, between being able to make a reconstruction that can perform something within a very specific experimental context and something that is nuanced enough to capture real life and be able to work in real life. So say a hippocampal prosthesis that works in a real life situation instead of a hippocampal prosthesis that works for a specific visual task.
Right.
Yeah.
A hippocampal prosthesis that only allows you to solve one task would be entirely useless.
Mm-hmm. Now, with this insight, what do you think that means for, say, the very popular attempt right now to try to build AI companies based around neuroscience insights, or the hope that one can take insights from neuroscience to create successful artificial intelligence companies?
I don't know. The question is, how informative is neuroscience at the moment for building state-of-the-art algorithms? There's this interesting contrast where you could say, what have we actually taken from neuroscience that currently makes it into into machine learning. Well, kind of the idea that we have many units that compute, that they're like locally simple, that like combining them allows us to do interesting complex functions. Maybe things like the concept of ReLU, but like what we took is very, very small. So I think the opposite is happening. I think what's really happening is that neuroscientists that work on machine learning problems are given a little bit of freedom by the world, where it's like, if I build something and I'm like, I think that that's how the brain does it, I will not immediately have to face that someone's using support vector machines and they get 1% more performance out of it. They'll be like, wow, that's really interesting. That could be meaningful about understanding the brain. And by the time I have finished thinking through that, my algorithm might be better than anyone else's algorithm. But it's, as a neuroscientist, I'm given that freedom to think. And if you look at artificial intelligence, machine learning, a good part of all the big inventions have been done by neuroscientists. How's that possible? There's so many computer scientists that dominate those fields. Why is it that neuroscientists contribute so much? Well, maybe it is, A, they have a source of inspiration, but B, they have that this freedom of thinking without having to immediately compete on something. And I think that is a strong driver of progress in the field.
This almost sounds like you're speaking from the pitch deck of Vicarious Inc., if you know that company.
I do know of Vicarious. I don't know why it would be on their pitch deck. Enlighten me.
It was way back in 2010 or 2011 when they were pitching to Founders Fund and others. Part of their pitch was, so Dilip George came up with this idea. He really wants to work on artificial general intelligence rather than artificial intelligence, meaning he doesn't want to train for or improve performance to a narrow goal. And because of that, he said, Vicarious will not be going after the hot topics, the hot goals, the things that everybody wants to solve, because if we have to do that, then we have to specialize early on in making our algorithm perform better than everyone else. And so then we won't be able to build something that is generally useful. So we want to pick the moderate target.
Right. But of course you can say early on. So the question is, what's the right model? Is the right model where you have a small number of companies like Vicarious giving themselves the freedom to think? Or is it maybe better if we have a relatively large number of small academic labs giving themselves the freedom to think? The question is, at this early stage, how much synergy is there and how much does it really help you to have a big team? And now, relative to any academic group, of course, Vicarious has a very big team. I don't know. I'm curious. Only time will tell how the different approaches will scale.
Yeah, that's always a really difficult question because, of course, back then in 2011, they did not have a big team. And you only end up getting a big team if you can attract enough investment, which you only get when you start going for the hot topics instead of the hot topics. So then everything changes, right? You know this from plenty of other startups that have to pivot because of the realities of the bottom line is what matters, right?
That's right, yeah.
Yeah.
Which is often said because the startups start with these beautiful, really big revolutionary ideas, and then in the end they improve Microsoft Excel or something, or Clippy.
It's absolutely true. So we were going through that one paper of yours, but it's interesting. I have another paper that you published recently that I thought was interesting, and maybe it even goes in a different direction. I don't know. This one is entitled Reverse Engineering Neural Networks from Many Partial Recordings.
Yeah.
Yes.
It goes into very different directions.
And I was thinking these directions are extremely interesting for people like, say, Ted Berger, who are doing their hippocampal prosthesis based on ePhys recordings from a very small number of sites over a short period of time. And so I was wondering what you think about that now, given the insights from that paper. And I realized that the neural networks in the paper that you were studying were fairly small.
So I think they get at very different questions. So what does it mean to understand a neural network, the paper we just discussed? ask at some level, how can we get at the insights that would be important for people if they want to feel that they can understand something? How to reconstruct a neural network from many partial recordings goes rather into the opposite direction, where it's say, I want to build a system that maybe approximates my brain or yours, and I can only have a certain budget of how many neurons I can record. So that model, that paper for reconstructing neural networks asks how I can use very big data sources to produce good models that we cannot understand. No, it would be if I take lots of recordings of maybe I can record from your brain 1,000 neurons at a time, and I'm asking how can I allocate my recordings so that I have the best simulation for you at the end. In that case, I want to say, well, what do I want to do? Do I want to take like a small subset of neurons and get lots of data from them? Or maybe I want to get like 1 second where I record 1,000 of them, maybe in the early visual areas, and then 1,000 at the next layer. Or maybe I want to take like random subsets of 1,000. It is in that space where it says, if I can record, how should I record so that I basically understand the function implemented by it? But if I say understand here, I just mean that I can approximate it. Now, I want to be able, in that paper, I want to be able to build it, something that approximates it. Mm-hmm. Whereas in the other case, I want to take the data from the brain, and I want to be able to call you afterwards and say, hey, look, I understand how the brain works. Let's spend 10 hours and I'm going to explain it to you and you'll be enlightened. That second paper in no way aims towards that. Now, like the first paper, if we'd be successful about brains, we argue that it's almost like you should replicate the neural network code that makes brains. Whereas in the other paper, we're trying to approximate the function that is a brain.
Yes. I agree completely with you on this distinction, because that's the distinction I draw between an emulation and a simulation.
Yes. So for that second one, like an emulation, we don't need to understand why it works the way it does. We just need to correctly do it. And so in the first paper, what does it mean to understand a neural network? We basically said, how could we get a description of the brain that is short enough that we could write it into a book? Yeah. When it comes to emulating a system, I don't care that the weights can be written into a book. There can be 1 billion of them, as long as it emulates the right system. Yeah. If it talks like Konrad in the end, makes the same bad jokes as Konrad, it's a success. And in that sense, those two papers really get at fundamentally different things.
Yeah. And what do you think your paper said about, say, the chances that something like the Berger approach to a prosthesis has?
So I don't know. So the Berger approach towards prosthesis, I think a lot of it boils down to, okay, so hold on, let's take apart the two things. One is what I mean about Berger's approach, and the second one is what it takes to be able to reconstruct an I/O system. So if I view hippocampus as basically a system, it has an input, it has an output, we want to make it so that it has the right outputs for the right inputs. The question fundamentally is, how complex is this function? And then the other part of the question would be, now, how many recordings do I need to be able to approximate that function? So from playing with these neural networks, as soon as there's really interesting questions involved, you need a very large number of experiments that you run on it. I don't think today's technology, certainly not what Berger used, is in the domain that it would require to really discover any truly interesting functions. And indeed, how are you thinking about hippocampus? I mean, if we believe that it somehow implements the the memories that we have, if it somehow allows us to, say, link our timeline into the things that actually happened, then it must be a system that is at least moderately, probably very complicated, because it needs to say provide pointers to the events that happened in that period of time, and there's probably a good number of them. So from that perspective, if you ask me to speculate on how complex hippocampus is, I think hippocampus is rather impressively complicated. And in that sense, directly as a corollary of that comes that we need lots and lots of recordings to reverse engineer it. I haven't seen data from that shows that these prosthetic devices get into the space where they could meaningfully approximate a system.
That's probably still the state we're in right now.
And I mean, time will tell. Maybe maybe in a few years' time, we will see lots of people have prosthetic devices that actually solve those problems. But let's just say I will be very surprised. But look, if it works, it would just be spectacular, giving back memory to countless people. That would be the best thing that neuroscience ever gave us.
Mm-hmm. Yeah. Okay, we could take this as a way to start talking about why a different approach to whole brain emulation is particularly interesting. But just before I do that, I just have to round off with this one paper because the last two I mentioned are a bit newer, but they never received as much publicity as the Could a Neuroscientist Understand a Microprocessor paper that Eric Jonas and you wrote together. So, what would be your principal recommendations to neuroscientists based on those findings?
So let's briefly maybe talk about what the microprocessor paper was about. Just the microprocessor paper, we basically, look, a big problem in neuroscience is we don't know if our approaches actually work, because if I say something wrong about the brain, it has no way of defending itself. So what we did is we basically took a system that we understand, which is the microprocessor, the 6502, and we asked, if we did the kind of experiments that neuroscientists do on brains, how enlightening would that be about the microprocessor? What was shocking throughout the paper is our results often look very similar to the way real brain results look like, and the insights are largely vacuous. So that paper, in a way, set me on a trajectory of asking, what does it mean for neuroscience to meaningfully make progress? So the paper, for example, what does it mean to understand a neural network, is a way of making the problems that the microprocessor paper showed super concrete. Now we can basically say, okay, there's this kind of complexity, this kind of complexity produces the following set of problems, and that's why we would have trouble really understanding a brain. But the microprocessor paper started it, and it fills in the fabric of neuroscience a very unique spot. You can say it's epistemology. Now, epistemology is about what we can know or what we can learn based on kind of experiments that we make. And what we found there is that there must be something wrong about the logic that we use in neuroscience. And based on that, what we recommend to neuroscientists is we have to be clear about the end game. So people like writing little papers. Every field has their standards. Not like if you're in neural recordings in some cortical area, it has its standard. And if you do functional connectivity on fMRI signals, it has its standard, and so forth. Lots of people work according to the standards. But there are very few people who ask, well, if the field does any number of experiments according to the standards of the field, where does that bring us? Where does that lead us? And the microprocessor paper, in a way, showed that the standard simple approaches that whole parts of the field are doing, in a way, don't really get us anywhere.
Yeah. So then your recommendation on that would be?
The recommendation is we need to ask ourselves what we are aiming for. And that is exactly what does it mean to understand a neural network does. It basically says, look, how every neuron interacts with every other neuron, and alternatively, what the tuning curve is of every neuron must be a question that we cannot answer, because this is where the information from the world sits. Therefore, at best, we can hope to understand the things that the brain optimizes, maybe how it optimizes it, maybe how it initializes itself, maybe how it sets up its architecture. Those things we can hope to understand, but we shouldn't hope to understand, say, the results of connectomics or the results of tuning curve mapping. It will have to be, if the brain has anything like a neural network where it basically soaks up information from the environment, it will have to be impossible for us to understand it. And it suggests that we should be doing neuroscience very differently.
Okay, this does lead interestingly into the next bit, because you just said that, hoping to fully understand connectomics, to get from that what is going on in a part of the brain, that might be impossible. I wanted to pose this next question to you, which is much more complicated really, but it is definitely directly towards whole brain emulation. In this little blurb that I wrote about where do we go from here, I was imagining a future where, say, by 2040, we'd be able to, connectomics, doing the complete mapping at 5 nanometer scale of something like a Drosophila brain would be no problem. And it would make visible not just the spine and the synapses, but even a number of other interesting molecules and objects that had been captured within that system that was preserved somehow, that was made available to the scan. And I imagine a future in 2040 where, doing this sewing machine of recording probes that Neuralink has embarked upon with Philip Sabies is a reality, and maybe even much more advanced than that, so you can record from a very substantial part of the brain through whatever technology. So if we just think about that for a moment, I'll set this out by first saying that when we talk about whole brain emulation or any kind of reverse engineering like this, we often contrast two different approaches. The one is the one that is based largely on what you know from, say, the Berger approach, or what you did in your paper there with your artificial neural network. You record a lot and then you try to reverse engineer from the recordings what is there. The alternative approach is that you take the detailed three-dimensional structure that you get from the connectome, the synaptome, etc., and you try to imbue that with function by, say, looking at the width of your dendritic pieces and trying to come up with the right membrane capacitance and resistance for that, and then put it into a big model, and then add to it additional information that you have about the types of receptors that you've managed to identify there, and etc., so that you get from a structural model to a functional model. And this is something that I think hasn't been done much at all. Maybe with the exception of some work on C. elegans, even Genelia Farms is only just starting to look at how they could even reconstruct some visual neurons in the Drosophila from this connectomics approach. But this is the approach that seems like the sort of thing where you could handle the kind of scale that you come across if, say, you want to do a whole brain emulation of a Drosophila brain. Let's just think about the Drosophila brain, not even the human brain. So you want to do a whole brain emulation of a Drosophila brain, and you think, if I can scan the connectome very carefully, and if I know a lot about the neurons in the Drosophila brain, maybe I can assign functions to all of this, and I can reconstruct this Drosophila brain.
Great.
So I guess my question to you then is, if you think about this approach and you think you're in the year 2040, what do you think is the biggest problem that you encounter, of course, as someone who knows a lot about models, and how would you proceed if you were going to try to come up with the next, say, R&D steps to get further with that study, to overcome these problems?
Okay, so let's first delineate the question a little from what we discussed before. So imagine I had a simulation. Let's say I have a Drosophila brain. It's cut into thin slices. I get all information out. I simulate it, and it works in the sense that it behaves just like a Drosophila. Mm-hmm. at some level, from an emulation perspective, we succeeded. Does that mean I really understand it? Arguably not, but that's not what's necessary to solve the engineering problem of emulation. Like, you can say, if we play that game, if we emulate it successfully, we have succeeded. It doesn't matter if we understand it. In fact, it could be that all the synapses that are there are just of a nature and complexity and basically represent all kinds of things that Drosophila learned over evolutionary timescale that, like, you or me will never possibly understand it. Doesn't matter. We successfully emulate it. So if we then so in that sense, like, it's really a different problem, and it is a much easier problem to emulate than to understand the system. And when it comes to emulation, let's talk a little about the path that one would need to take. So methods like connectomics are great in the sense that they give you crazy amounts of data, like really unbelievably, ridiculously crazy amounts of data. And so far they're weak in the sense that arguably what we want to reach is an emulation of the physiological processes. Like, we want the neurons to have the same output. We don't want the neurons to have the same form, because that would be something that there wouldn't be success at what we're after. So if we want the neurons to have the same output, we need to, and we want to use the big data that we could get from connectomics. We need to have that translation layer in place. Basically, give me the connectomics data now, I can tell you what the neurons' weights and nonlinearities and all things like that are. Now, there's of course I should also mention there's the hybrid approach, where you record from lots of them, and you also have the connectomics. But let's say it's possible at the moment. I haven't seen anything that convinces me that it's not true, that it is possible, if we say have connectomics together, maybe with like molecular characterization, maybe expansion microscopy, maybe look at all the different protein species, you name it, that from there we could get a perfect or near perfect translation into an electrophysiological system. No? And we can formulate that at the level of a single cell. We can do it at a single cell, we can do it at the whole brain. So if I give you a whole cell with all its inputs, it's still a lot of them, and all its outputs and all the proteins that I can detect, and like the dendrite reconstructed at the best resolution and so forth, can I now know what the neuron's output will be as a function of its input? If we could solve that problem, it would be rather obvious that we could be, or like it would seem very likely that we can be doing meaningful whole brain emulation. Now, that task may be extremely difficult, and just relating that to recording, it's not clear. Let's go to a very different scenario here. Imagine you could record every neuron in a brain, every single neuron at perfect temporal resolution. It's unclear if you could reverse engineer what a brain does then. And the reason for that is the following. There's only a certain time frame over which you could get that information. And maybe the time that it takes, like neurons on average have like 3 spikes per second. That's, I believe, don't quote me, I might be wrong by a factor 3 or something. Yeah, but like imagine it depends on the type of neuron. But imagine that gives you like 10,000 spikes per hour, that's like 200,000 spikes per day. That is still a relatively limited amount of information, 200,000 spikes. If cortical neurons have like a couple thousand inputs, so that would give us a relatively small number of spikes per input. So within a day, if neurons were linear, we might be kind of able to identify what a neuron does, but it would be better if we had 100 or 1,000 days for that. But if neurons were nonlinear, then no amount of recording time, unless they're in a very small class, but if there's really a lot of nonlinear aspects about the neurons that we don't know, then no amount of recording can tell you what a neuron is. neurons. So in that sense, like connectomics seems to potentially give us much more information about the neurons than we could get by having basically lifelong recordings from all neurons. And lifelong recordings wouldn't be enough because you learn things.
Okay. So just very briefly before we get into more about this, just what about if you had recording and stimulation?
So if you had recordings and stimulations, you would buy very, very little. So if you can record from all neurons, you see along with each neuron all its neighboring neurons. And in that sense, you know the input, you know what the input output, you can at least hope to be able to identify the input output function. Now, so when it comes to causal inference, we can be more precise there. There is the so-called backdoor criterion, which means that if I have a causal system, maybe like internal stuff of a neuron going to the outside, and I basically have a set of other recorded things that entirely separate that from the other variables in my causal system, I can identify what the system is. Mm-hmm. So if you can record absolutely all neurons, it really largely boils down to identify each neuron based on like some vicinity of it. That's like a big if. And if we could stimulate it, we can do things better. Now we can make sure that they're actually independent. We can make sure that they're white. So one problem for identification is that that if we look at the dimensions, there will be some dimensions that are associated with very high variance, maybe like all neurons are more active or all neurons are less active. So it's very easy to identify what neurons do with respect to those dimensions, whereas there will be other dimensions, maybe two neurons that are very heavily correlated, where one is a little more active, the other one is less active. Those dimensions are very hard to identify them. And if we could stimulate, we can effectively make things white, and we can make a lot of the causal inference problems go away. But it still doesn't change the basic calculation that the spikes you will see in the lifetime of a neuron might not be enough to identify the neurons if they were truly nonlinear. Yeah. So I don't think that stimulation is really going to solve those problems. But stimulation can make it much better. There's no doubt there. Mm-hmm. And again, a lot of it boils down to which level we may expect or hope that there's something that is simple about the system. So some people say that brains don't consist of 10 to the 10-ish largely independent neurons that all do really complicated things, but where basically everything that's computed isn't computed by one neuron, but by a large group of neurons, maybe 10,000 neurons. They just jointly compute that one thing. In that case, well, like all of a sudden you're a factor 10,000 lower in the amount of information you need to describe, and everything gets to be much, much simpler. I don't think I've seen any evidence for such an idea of very low-dimensional circuitry in the brain. And quite possibly, if our brains were truly low-dimensional, we wouldn't be having the level of discussion we're having right now.
Indeed, yeah.
So basically, if it's high-dimensional, then it's unclear how much stimulation helps us, and it's unclear if any amount of stimulation and recording can actually identify a whole brain, a whole human-scale brain, and maybe no amount of stimulation could identify a Drosophila brain. Mm-hmm. Okay. Whereas if we could solve that inverse problem, that translation problem, where like, let's say you give me all the proteins I have in a neuron, and you give me like the whole membrane, how it's formed and so forth, and somehow we can solve the problem of translating to physiology, that might be a much more handleable problem. It might not be. I mean, like, basically, the future will have to tell. Yeah. But it's possible if the brain is actually complicated, then no amount of recording and stimulation can solve it. And it's possible that that for some reason that translation problem of like basically give me like maximal connectomics style data and all the proteins and everything, it still isn't sufficient, in which case we'd be running into a dead end.
Yeah. So I was going to ask you to imagine as a modeler if you were doing the inverse problem of translating from this connectomics information to a functional model, where you think that this model will go off the rails. But maybe I'm also very interested in what you mean when you say it is possible that this inverse problem is not doable, and I'd really like to know if you think that there might be a fundamental reason, it'd be very interesting to know what you think that might be, unless it's a general hedge.
It shouldn't be a general hedge. That would be bad style. So let's be precise about that. So there exists some information in. So a cell has undoubtedly a lot of information. Some of it might be in how big the synapses are. Some of it might be in what proteins are where on the synapse. Some things might be in terms of how a specific protein has been modified at the synapse, and some of it might be in some bizarre configurational property of the protein. It's possible that basically you need ongoing maintenance of the property of the proteins or whatever it is that carries that information, that if as soon as you start measuring, it's that the measurement process destroys the things that you want to measure. If that is the case, there could be a lot of scenarios where things are fundamentally impossible. Like we can't simultaneously measure momentum and position of something. Right. So that comes from physics. There may be chemical analogs about that. Say, if I want to know which protein there is, maybe I have to destroy the protein and can no longer see its shape. Yeah. Those are the spaces in which I see potential destroying block stuff that we could never overcome. But in principle, and that's why I find it sad that there's so little activity. Like we start having connectomics, we start having morphologies of lots of neurons, and yet it kind of doesn't even exist as a research field. Like, why is it not that we have like a solid field that says, okay, let's take cells, let's take all the measurements that we have, let's see how much we can bridge between them. Instead, we like go crazy on like starting to do. It's beautiful, but like basically we start to do connectomics on like reasonably big systems instead of saying, okay, let's take a small system and see if, based on the granularity we're getting at the moment, we can solve that translation problem. And there's very little research in that area at the moment, and it's a little sad.
Well, I think a part of the problem is that the majority of the people working in connectomics aren't necessarily interested in the translation problem. They're happy enough with the neuroanatomical information that they're getting.
Yeah, but if we want to evaluate to which level it can inform us in thinking functionally about brains, why is connectomics important from a health perspective? In large part, it's because it affects the way the brain computes potentially, because it affects the way the the brain is affected by diseases. So if we don't build that bridge level in between, it's really just stamp collecting.
Yeah, that's true. And I think that computationalists who like to build functional models can be made to see that. It's a little harder with people who are used to looking at data and making correlations with diseases. They'll see.
Yeah. That is unfortunately a little bit the state of the field. And in my lab is getting very much interested in morphologies these days, which sounds quaint. It sounds like it should be a 19th century thing. but how can we actually describe morphologies? How can we see structure in morphologies? It's a deep set of questions.
So speaking of questions, I did want to get back to that question about how you think that trying to do that translation, even if, let's say, we don't have a measurement problem, let's say it's fundamentally not impossible to get the data that you would need to solve the translation problem, how could modeling based on that still go off the rails? What are the things you have to watch out for? And I'll just give you an example from the top of my head. that because we can't measure everything perfectly, and there are always some approximations involved in how we build this translation, that it's possible that unless the homeostatic processes of your system pop into being as part of what you're implementing, that perhaps then the cumulative error might cause the system not to perform the way that you want to. That's one of the things I worry about.
Right. Yeah, no, there are all those failure modes. It may be that we have trouble at predicting dimensions that are important, but that don't have a lot of variance. It may be that we have difficulty that solving. So in machine learning, it's much easier to be good at predicting something than being good at understanding why you're good at predicting something. So it will be hard to kind of like have a model that doesn't just replicate the output, but that replicates the true input-output function. Now, it could, for example, be I use one set of stimuli, and for that set of stimuli, I'm really good at predicting what the neuron will do based on its morphology. But as soon as I leave that set of stimuli, will be as a chance. Yeah. In that case, I would have fooled myself believing that we're good at it. Mm-hmm. No, and so you could say we might have, let's say, a person and a simulated person that have like exactly the same object recognition. They both say, well, that's a dog. But they could internally be very, very different because we've been using the task of dog recognition to calibrate the system.
Right. So,
and therefore it wouldn't generalize.
Calibrating or validating a system, that's a very big question, of course. How would you do that for something this large? Even for Drosophila. How you run
but for Drosophila, you can say asking for generalization is good. No? Like you could say, let's train a model on Drosophila exclusively in like a vision prom, and let's see if it will fight other Drosophila. Mm-hmm. You can ask for those generalization tasks. The question is how good it can be. And now, if we train it on all possible behaviors, well, in a way, we just build in that it can produce all behaviors.
Right. Yeah.
Anyways, it's an important set of problems, and in that sense, like I like people who think about brain emulation because it's a clear endpoint. It's clear what it means to succeed, and it's pretty clear what it means to fail at it. And that is a problem that, as I mentioned earlier in our discussion, that in neuroscience we often don't know what it means to succeed or fail at something.
Mm-hmm. Yeah, that's interesting. This is a question that Ken Hayworth thinks about a lot because he feels like neuroscientists should sometimes step back and see where is the end goal of neuroscience, what does it accomplish, and what should you aspire to.
I obviously couldn't agree with Ken any more than that.
Indeed, yeah. I have to actually end the interview here. I want to thank you very much for all your wonderful conversation. This was extremely enlightening to me, anyway, and also fun.
Yeah, thanks so much. This was great fun.
Many thanks to Professor Konrad Kording for that fantastic interview. Once again, I would like to remind our audience, if you have any questions for our panel discussion, please post them into the chat. We'll right now go into our second interview recording. Our second interview is with Professor Dong Song of the University of Southern California. And right after this interview, we will begin our panel discussion following this interview.
And I was wondering, given your recent papers that have really gone into two very different approaches, you've used the large-scale model and you've used the so-called MIMO model. Could you say a few things about those two types of modeling and the best situations where you think you would apply one or the other?
Sure. So, yeah, actually, I just submitted a proposal. I propose to work on exactly like this, how to use two approaches, right? One is what I call mechanistic model, right? meaning using a lot of mechanisms, using all the knowledge we have about the biological system. And sometimes when the knowledge is missing, we use hypotheses, right? And also pieces of information from different sources. And use this kind of data to build what I call mechanistic, or some people call that biological model, right? So I think this kind of model is really important for review how the underlying mechanism determine the function, right? Determine behavior. All the mechanisms work in concert, determine, and also the internal dynamics of the nervous system, right? So that's really important. And nothing can replace this kind of modeling, because this is the most direct biological modeling, right? But on the other hand, what I call input-output model is more like the currently popular machine learning model, right? It's a purely data-driven input-output sort of mapping, right? And that's very useful in a lot of applications, such as like machine learning, classification, and also in all specific approach, doing like cortical processes, right? In order to interact with the brain, we have to work with the data, right? And use the input-output data, right? Without complete knowledge about the underlying mechanism, just to try to do the best we can to predict what output is based on the input, right? That's a very useful approach. So my belief is that I don't have a strong preference on either of the modeling, right? Because I do both from very beginning. My PhD thesis is on parametric and non-parametric modeling of the synaptic transmission, right? And in that context, parametric and non-parametric actually means mechanistic and input-output modeling, right? But now later I realized that non-parametric or parametric modeling in statistics, they mean very different things. So I changed to mechanistic and input-output model. So my belief is always you have to combine those two modeling approaches together to better review how the biological system works, especially the brain.
Yeah, did you know that Konrad Kording recently co-authored a paper titled Reverse Engineering Neural Networks from Many Partial Recordings? Had you seen that one?
No.
It's a simulation study where what they did is they created a number of neural networks, and then they pretended that they were able to record from some subset of the neurons there, and then try to reconstruct the function of the network, sort of similar to the MIMO method, I suppose, but a little different because they have full control over the underlying system. And
so what the result like? I'm assuming it's like extension of his whether a neurobiologist can build a computer, right?
It was somewhat related to that, because there was also another paper he published, or is publishing this year, in which he's looking at what it means to understand a neural network. Although he really looks at this as two very different things. So, for example, when he talks about how to understand a neural network, he's trying to make the case that that we may be trying too hard to understand in terms of, this is a neuron that is tuned to seeing edges. This is a Jennifer Aniston neuron. This is a region that does this. And instead to understand in a different way, that you would understand neural networks, because neural networks are learning things, and you want to understand the way that they learn and the type of information they learn from, and then the rest is very malleable. So that's what he says about understanding neural networks. But on the other side, when he says you're trying to recreate what a specific network is doing, like a patient's brain, or part of a patient's brain, or even an artificial network, that trying to understand that is a different question. And it's not so much that you're understanding it, it's that you're trying to replicate what it's doing, even if you don't entirely understand what it's doing. And that's what this paper about the partial recordings was more about. And actually he was quite encouraged, because I think he was fairly critical when he was talking about the hippocampal model at times, but he was fairly encouraged to see how well they were able to reconstruct the function of the network in terms of the errors that it made and the performance that it achieved. even for a fairly small subset of samples.
Right.
But at the same time, when I had a conversation with him about it, he made it clear that he didn't really think that this could work as well for large networks with complex behavior. Yeah, and especially he said if it turns out that collections of neurons and the neurons within those collections behave like nonlinear functions, in which case it's very, very difficult to reconstruct what a set of nonlinear functions are doing from a few samples like that. What do you think about this, about his statements?
Yeah, but I don't know the context, right? So I'm not sure why he said nonlinear function is the key, right? Because as I see in machine learning, there's so much progress in mapping nonlinear functions, right? When you think about like pattern recognition, right? Recognize photos of an animal from different angles with different sizes, right?
Yeah.
That's a highly nonlinear mapping, right? And it's been solved beautifully, right, by deep neural network. So I'm not sure whether I agree on the nonlinear part, right? But I do agree, because input-output model usually have a pretty predefined structure, right? So we try to solve the input-output mapping. So the knowledge is somewhat limited by the structure itself.
Yeah.
Right. For example, if you have a linear model to fit input data, I don't think you can learn much about the nonlinear property. Right. If you have a Volterra form, like a second order, third order Volterra model, like Taylor expansion, that kind of mapping, I don't think you can go much beyond what third order polynomial can represent. Right.
Right.
So yeah. So I feel like the limitation is mainly in the structure of the model. But on the other hand, if we have a real universal learning machine that can map arbitrary nonlinear dynamical system, right, then the issue, then I sort of agree. It's probably I still won't say it's a nonlinearity itself, right? It's a complexity.
Yeah.
The computational principle of the system, right?
Right.
We need to make sure, because input-output model is still a tool, right? We need to make sure the tool is sophisticated enough to explore that kind of complexity of the brain.
And would you say, so I mean, the complexity of the model and whether it will converge and all of that sort of thing, that matters, but the data that you have matters too. Right? So if you're trying to describe a more complex system and you have a very limited amount of data to do it with, that could be a problem.
Right. Yeah. Definitely. The simplest example is that, so if you have a nonlinear function, right, you can, in your mind, you draw a nonlinear, right, non-straight line function. If you look at a very small range, that can always be approximated by a linear function, right? And if you go beyond, you make your input data broader, right? Broadband data, then you start to see the nonlinearity, right? And that's also true. That's a very simple example, but that's also true, for example, in our hippocampal memory persistence study, right? So what we proved, what we shown, I think is what we show is that in a specific patient, in two specific brain regions, right? When we use the code, we can partially restore, or sometimes even enhance the memory function specific to that memory task.
Right.
So there could be an overfitting to that specific memory task, although I don't believe we overfit the data in that particular model. Right? So,
Yeah. Okay. I think that lines up with his thinking as well as mine, and it just makes sense, I think. If we can zoom out a little bit, let's back up from this very detailed discussion about which kind of model to use. In your opinion, what do you think is success for neuroscience? So what is a successful outcome if you think about neuroscience in the long term? What is it really there for? Why are we doing it?
So first is curiosity, right? Brain is still largely a mystery. So we need to understand all the fundamental principles of the brain, right? we understand, computational principle in the sense of the David Mark's definition, right? He defines 3 levels of understanding to the brain, and the top level is the computational principle, right? What the brain region is trying to do, and below that, what kind of algorithm is used, and in the end, what kind of mechanism is involved. So I think that's still, to me, the most important goal of neuroscience, right?
Yeah.
Because even only given that kind of knowledge, we can start to think about how to fully, fully understand how the brain works and how to build a machine that can completely replicate the brain function, right? So that's always the ultimate goal, I think.
Yeah, so on the one hand, it's pure curiosity, but the pure curiosity very quickly moves into more applied curiosity in the sense of, can we build an artificial machine that does something like this? Can we build a prosthesis? All that sort of thing, right? Yeah. Okay, I see that.
Yeah, I forgot who said that, but I remember somebody said that, you fully understand the system until you can build one, right?
Right, yes. So, that was Richard Feynman, perhaps? Possibly?
I don't remember.
Yeah, it could be that it was a Feynman quote, but maybe it was someone else.
Yeah.
Okay, so then a prosthesis or an artificial brain, because artificial brains, of course, might be entirely separate from prosthesis. It could be an AI, but maybe built upon principles that we learn from the brain. What do you think, how does one go about validating models that you use for either a neural prosthesis or some artificial brain that you're building based on these principles? How do you validate whether you're going in the right direction?
So in the case of prosthesis, we did the validation in multiple levels, right? The first is the lower level. We want to make sure, because we took an input-output modeling approach.
Yeah.
So first, we want to make sure we are not overfitting the input-output data we got, right? The model has predictive power in terms of replicating the output signal, right? So, and there's a lot of mathematical tools for doing that, such like cross-validation, right? You always try to validate your model in terms of a new data set. You use the model estimate with a different, completely independent data set, right?
Yeah.
So by doing that, you can make sure you are revealing the underlying structure in the input-output model. Right? You are not fitting noise or something just happened randomly, right? You got some level of generalization of the model, but obviously that's not sufficient, right? Because even you can fully replicate what the model, fully replicate what the input-output data you recorded. That's very important.
Yes.
It's not the full data set, right? You can always record a very small subset of all the neural activity, right? So you need to make sure several things. First, that small subpopulation is large enough for whatever you are trying to do, right? For example, whether that's enough for replicating the sort of memory we try to study, right? Maybe for some complicated memory, it's not enough. Like I don't think we ever recorded Bill Clinton's cell. Right?
Okay.
So I don't think we can ever predict whether the person is trying to remember him, right? But on the other hand, right, if you look at like spatial representation, now if you would go to animals, everybody, every neuroscientist, they will agree, if you record 30, 40 neurons, very likely you can tell where the animal is, right, based on the place field firing, right?
Yeah.
So that's another extreme case. It really depends on what kind of behavioral task you are talking about, so that's so you're right. You need to make sure, you need to validate your model in terms of whether you can replicate that kind of that level of memory representation.
So the validation is directly related to the goal of what you're building this thing for.
Right. Yeah. For example, in one of my earlier paper. So we built a MIMO model, right? And on top of building MIMO model, we also did the memory decoding on the predicted output signal, right? Yeah. We try to make sure from the predicted output signal, we can classify the animal is pressing the left lever or the right lever, right? It's a very so-called binary memory, but we need to make sure, although we claim, right, the prediction is really accurate, but that predicted output should be sufficient for decoding those two binary memory, right?
Yes.
So I see that as another level of the validation, right? This is related to the behavior, related to the memory, but it still largely depends on the data itself, right? It's not a behavior yet, right? And then that leads to the next level, which is the most important level, I think. It's whether the prosthesis can really restore function, right? When you need to design specific behavioral tasks, you need to have a way of to quantifying memory function and to show using this approach. And you need to have very well designed negative control, right? You need to show this approach can significantly restore or enhance memory functions. Right?
Yeah, indeed.
Yeah.
Okay.
And actually, there could be another layer in between, which we don't have access to. We don't have the technology to record yet, right? But I think that could be potentially really important. It's whether we can, if we can, back up a little bit before we go to behavior, right? But after we make sure we can predict the output signal, we need to go to another brain region, like further downstream in the signal transmission, to monitor as many neurons as possible in that region, to make sure whatever simulation we generated is generating the appropriate neural dynamics in that region. Right? That can be an important intermediate step before we go into the behavior. Because behavior is too far away, right? It's very far away. And also behavior, there's a lot of limitation in terms of behavior. When we go to hospital, you are not allowed to interact with the patient forever, right? You only got a few hours. So you have very sparse data about testing the behavior. So the intermediate step, right, for testing, for recording the neural dynamic could also be extremely important.
Yeah. And testing the intermediate steps, like testing different regions and actually looking at the effect on recordings there, that could also be a way to substantiate that that the subjective internal perception of what's going on is probably similar. Because otherwise, if you're just looking at behavior, you could say the same behavior can be accomplished in many different ways, and the patient may be feeling something very different going on.
Right.
So that's another one. Okay, so I'm almost at the last question already. Can you believe it or not? And this one really asks you to kind of imagine yourself a little bit in the future, and we're thinking maybe 2040, around that time, or 2030, 2040, 2050. It's not so important exactly which time, because it on a large scale, a historic scale, this is all all tomorrow, basically, right? It doesn't really matter if it's a few decades or whatnot. Imagine that the technology we have to look at the structure of the brain improves as it has been going, and we can get very good maps of small animals by now, and even of pieces of the brain. And imagine also that our ability to record has gotten quite a bit better. First of all, I'd like to ask then, if it comes to when you're thinking about doing something like a prosthesis or an emulation of a brain or understanding the brain, and maybe these are different answers that you would give for those, do you think it would be better to try this first and test this with, say, a very small animal, something like a Drosophila or some other very tiny animal? Or would it be better to try this first, say in 2040, with all those technology that's there, with a portion of a human brain, like what you're trying now with the hippocampal prosthesis?
Yeah, I'd like to say we probably need to do both, right? So because we had conversation like this before. I know you have been interested in this full brain emulation for decades.
Yeah, absolutely.
Yeah. For doing that, I always believe starting from a small animal. I'm not saying at the same time you shouldn't consider human at all, right? But to show some solid example from lower animal would be really important, right?
Yeah.
Like from the Elegans or from some animal, you can have a recording of every neuron, right, using whatever technology available, right? Because a lot of neuroscientists say now I really want to record every neuron in the brain, right, in human, right? So that's the data I dream of, right?
Yes.
And in some lower animals, now you already have that, right? So whether that's enough for you to do whatever you are trying to do, right? That's a very good example, very good starting point, right? So you have, the full record of activity. You have all the knowledge about the connectivity, right? You can even study electrophysiologically how each neuron behaves. So that should allow you to, faithfully reconstruct this neuronal network and hopefully replicate all the behavior, right?
So you think that the technology to do that, does that exist today, or do you think that exists in 2040 if you want to do that with a very small animal, like C. elegans or Drosophila? Let's say someone really wants to do that project.
So if it's not already ready, I believe that should be ready in decades, right? It shouldn't be in far future. People are already doing that, are already trying to do that, right? So,
yeah, certainly for C. elegans, there are some attempts,
right.
But
on the other hand, I think people obviously still need to continually working with human, right? For a variety of reasons. Because the reason, for example, we build hippocampal memory prosthesis, it's not really for replicating the complete function of the hippocampus, right? So what we try to do is not from 1 to a million. What we try to do is basically from 0 to 1, right? Because in some of the patients, because of a lot of the serious neurological disorders, the hippocampus can be completely removed, right? And in that case, I'll be happy to help those patients from cannot remember anything at all to remember a binary information.
Yes.
Right.
Yeah.
So, and that's very, very. So even we can do that, right? And I believe we already showed that. Even we did that, that's very, very far away from completely understand the hippocampal function or build the artificial hippocampus.
Sure. Of course.
So,
yeah.
Yeah.
Yeah. So now, if let's say that that you wanted to create an emulation of a Drosophila in 2040. How, if you were starting this project, where would you begin? Would you begin with something, let's say, that is entirely based on doing a lot of recordings and then trying to do something that is more of a statistical approach, or would you do this more like your large-scale model? How would you go about it if you were to start this?
Yeah. So I guess I could be biased by the proposal I just submitted.
Okay.
I really like to do a full-scale model and recording of some animal, right? If that's possible. Maybe starting from a lower animal like C. elegans, or at some point maybe mouse is with the advance of the like optogenetics, right? All the imaging techniques. Maybe one day we can record all the neuron activity from single mouse, right?
Yeah, indeed.
And what I really want to do is get that kind of recording, explore the animal with a lot of different behaviors, and build a full-scale model, right, of a certain brain region, such as hippocampus, to see whether we can, fully replicate the behavior and replicate how it remembers things, how it navigates in space.
So is this what you'd like to be doing in 2040?
In the context of the emulating the brain, right? But but on the other hand, I also have a strong interest in, if you think about it, you don't use the word prosthesis at all, right? You don't talk about replicating brain function. You just think about the deep brain stimulation.
Yeah.
Right. So using certain, electrical or whatever intervention method, try to influence the brain's behavior try to enhance brain function, right? When you think about along that line, right, that will change what you want to do. There are a lot of practical things you worry about, right?
Right.
There are a lot of people we still don't believe you can actually using DBS to write in cognition or memory related information. They only consider DBS as just a way of modulating the brain function, right?
Yeah.
So there are a lot of issues over there. I'm not sure which one is more interesting to me because I'm doing both, right?
Yep.
But as I just said, right, maybe I'm biased by the proposal I just submitted. So at least for now, that's I believe that's probably more exciting than do a more refined form of DBS, right?
Okay. Great.
Yeah.
Great. Yeah, that was it for me. Thank you very, very much for giving me all these wonderful answers.
Many thanks to Professor Dong-Son for that fantastic interview. With this conclusion of our interview recording section, we will now begin our live panel discussion section. We will begin with questions that we have prepared at the moment for the panel. Then we will open up the discussion to our worldwide audience. Once again, our workshop is participatory and is open to all of our international attendees and viewers. You can ask questions, and when it is your turn, you can opt to engage directly with our panel, or you can leave it to the moderator to repeat your question aloud. You can write questions directly into the chat that is enabled for our YouTube livestream chat. One of our excellent staff members is monitoring the chat and will alert me to interesting and topical questions, which I will read to the panel. More complicated or multi-part questions, where it is important to establish that the question is properly understood and answered, are best asked directly by engaging with the panelists. We make this possible through our call-in number. The number and PIN are shown at the bottom of the screen right now. They are also written in the livestream description and on the livestream carboncopies.org webpage. They are 786-540-5631 with PIN 180-618-986. If you're calling from outside the U.S., you might also use the web call-in option at call.carboncopies.org to avoid telephone charges. The process is similar to calling in on a radio show. Your call will be monitored and a respectful exchange is expected. When your question would benefit from direct interaction, I will invite you to call in if you can. If you want to add a note to your question stating that you can call in or that you cannot, that can be very helpful to our moderator. And with that, we have some questions prepared at the moment by Dr. Randal Koene. Dr. Koene, would you like to take the floor?
Can you hear me right now?
Yes.
Okay. Well, so first of all, I want to apologize briefly to Dong-Song and anyone else who was listening to the live stream. Unfortunately, there's something like a 15-second delay between the version that is appearing on the Google Meet session that we're using to produce this event and its live stream. And as a consequence, sometimes it's easy to make simple mistakes. For instance, I think Michael thought that Dong-Song's video was already over because it had ended on his end, but it was still streaming out on the live stream. And so he started telling us what was coming up next while that was still playing. These sorts of things will happen until we figure out how to do this even more smoothly. And one part of that is we're going to have to figure out how to stream the live stream output back into the Meet session in some way so that the participants who are here on the panel can see that without having to switch to a different window with the live stream there. So this is kind of just a technical thing, but we're still trying to figure out how to solve that. Anyhow, thank you very much, everyone, for your contributions, especially Professor Konrad Kording and Professor Dong-Song, who provided these wonderful interviews to us. And I wanted to kind of launch into the conversation by asking a very, it's simple but maybe not so simple question. We've talked a lot about how to emulate brains and what the right way is to do that versus understanding the brain, or what sort of systems we might explore, say Drosophila brain. I think an important question to ask right up front is, why do we not yet have at least some kind of working emulation, a wonderful working emulation of C. elegans at this point And I wonder if any of you have an idea about that, and especially Professor Kording and Professor Song. Also, someone here still has, let's see, if you're not speaking, it would be great if you can mute yourself on the channel. For people calling in by phone, that's not an issue, but anyone who's on the channel differently, like Michael, you could mute yourself when you're not speaking. Thanks. So why do we not yet have a C. elegans emulation?
So, hey, it's Konrad speaking here. So I think the reason why we don't have C. elegans emulation isn't because people don't want to do it. I think the reason is because it doesn't work. So when it comes to C. elegans, it seems easy, no? It's like 300 some neurons. You can list them all. They all have names. We all have ideas on what they mean. The problem is that it's a very highly nonlinear system that communicates also along channels that you can't see. So it's largely analog. So it matters not just what each of the 300 neurons does, but maybe what each part of it does. And therefore, if people try to simulate C. elegans at the moment, it just doesn't work properly.
Mm-hmm. Okay. Now,
so do you, Konrad, do you think that that holds for other parts of the of mammalian brains? Like, do you think that the visual system is easier? I know you had a Twitter discussion with Michael Hendricks on this recently. Do you think that it's potentially easier to emulate, let's say, visual cortex, early visual cortex, than it would be to emulate C. elegans? Or do you think that we're perhaps fooling ourselves with thinking that V1 would be easier?
I think we're fooling ourselves if we believe that V1 is easier. So there are lots of factors that affect neurons in primary visual cortex. These neurons have elaborate dendritic trees. They have countless neuromodulators. We know that those neuromodulators are highly important. We know that it has, say, connections coming from higher areas and lower areas. And if you remove the higher layers, then bad things happen in primary visual cortex. So I do think that we are maybe overly optimistic about our current approaches. I think what gives me so much hope is that we start being able to really get really large data sets, and we haven't really pushed those approaches to the limit at all so far.
Yeah, but just to back up one tiny bit, if we just look only at the C. elegans problem for the moment and we say it's analog, it's nonlinear, there are approaches to attempting to map nonlinear functions. As Zhang Song pointed out in his interviews, he was mentioning, well, there are methods, right?
Yeah, so I would, this is Dong. I would ask about to ask this question to Konrad, is that you mentioned nonlinearity. Obviously that's a very important property of the neuron, right? So do you think the nonlinearity in the neuron is forbiddingly difficult to model? Because I thought, right, it's mathematically proved that if you have a multi-layer neural network, you can map arbitrary nonlinear function, right? So that's a universal, right, machine for modeling arbitrary nonlinearities. So why nonlinearity is the main factor here?
Well, the reason from my perspective why it's important is if a neuron is linear and a neuron has 1,000 inputs, we can reasonably hope in the period of an experiment. So for people to have a scale here, an experiment will maybe give us order 100,000 spikes from a neuron. So if we have 1,000 inputs and we do a typical experiment, we can hope to get 100-some spikes of information about each of them if we properly separate it. And then it gets to be a rather tractable problem when it comes to physiology only. Now, if it's nonlinear and the dendritic tree does truly important things and we can't see the dendritic tree, then for all that we know, any pair of synapses could be jointly able to, say, produce NMDA spikes, which is an intracellular phenomenon where basically if multiple inputs go to the same group of local synapses, they do something that is much bigger than the sum of the parts. And if these things do happen, then we would, in a way, need to go through all pairs of inputs and go through it. And all of a sudden, instead of mapping a 1,000-dimensional system, we have to map a 1,000,000-dimensional system, which will never be possible with 100,000 spikes. Now, I don't know where we are. That's the problem, and that's the sad state of neuroscience at the moment. I don't know how good we can be at describing neurons based on the assumption that it's basically linear with maybe minor nonlinearities, versus to which level these local nonlinearities are really important. If we knew, it could usually inform our endeavors. But if it is, imagine it is very nonlinear. It's well possible still that if we could, say, reconstruct the whole dendritic tree of a cell, not just have electrophysiology, that then the structure of it can maybe make the space of possible nonlinear functions that neurons do much smaller. So I really think that for the moment, we don't know where in that space we are, and those would be the first experiments that I think we should be doing, so that we at least know how we should think about it. And we haven't done these experiments in C. elegans either, just to coming back to Randal's original question. So basically, we don't know how complicated neurons are.
I see. Okay.
So, yeah, when I think about this input-output mapping, right, I often think about as a spatial temporal patterns of the input spike mapping to the output spike trend, right? So I agree with you, like we never have a long enough recording for exhaustively explore the input-output mapping for any neuron yet. But when you look at the input spatial temporal pattern, right, you think about at the time, one dimension, the number of neurons, which is, as you said, probably 1,000 to 1,000. But that's still not as complicated as natural image, right? So when people use machine learning to classify natural image, the dimension is even higher. And this can still very accurately predict the output, right? So I feel like, although it's complex, but it's probably not forbiddingly complex. At some point, if we get enough data, it's still possible to completely replicate the input-output mapping. So if we can do, yeah.
So let me just push back a little bit on that. So
okay,
let's take visual cortex. Do we know how much we know about it? Yes, we know that if we build a great predictive model, maybe we can describe half the variance. But if I predict that you'll be more active during daytime than at night, I will describe also a good part of the variance of, say, what your muscles are doing. But that doesn't mean that we're really good at understanding it. So I'm not sure that I can say, based on any data that I know, how well we understand primary visual cortex.
Yeah, there's this huge difference between being able to classify and being able to predict other possible outputs, so output that hasn't been seen in your data set. I mean, we have the learning set, we've got the validation set, we can train our models to do well for those, but this is no guarantee for anything else that you can come across. And I almost see this as being the main thing that you're currently arguing about, because yes, you can use a system like a neural network to learn how to map visual input and how to learn to reproduce the function of a piece of a system. But at the same time, you don't have a guarantee that you've captured everything about the underlying system. And so part of the argument is probably, in my mind, about what you would consider a successful emulation. At what point do you say that you've actually captured what you're trying to capture? And this, again, putting this back on C. elegans, the number of different things that a C. elegans network can do, if we just look at all the different outputs of those neurons and what they're driving, muscles, for instance, I can't imagine that is, I mean, I'm sure it's rather large, but I can't imagine that it's ridiculously large. I can't imagine that it's, approaching infinite. So it seems like something like C. elegans would be a system where even if there is nonlinearity present, and if we observed enough, that we could describe most of the behavior. Of course, I haven't really defined what most means, but that's how I see it. And so I'm still wondering why such a simple system as C. elegans hasn't been tackled. Because if we did, if we actually tackled that one, if someone had written up a paper where they described very clearly why it's impossible to tackle it, this would say a lot about emulating any parts of the brain. And if someone had written a paper where they actually showed that they could emulate C. elegans because they'd made enough measurements and then they predicted the system correctly, then that would also give us something useful. So I guess I'm a little bit surprised that it hasn't been done, and I'm almost beginning to think this has a lot to do with Konrad's complaints about what neuroscience is focusing on.
But come on, I mean, look, Eve Mader has, for her entire career, tried to understand a system that is factor 10 smaller effectively than C. elegans, which is the ganglion of the lobster. And you can say by every imaginable idea, it will be a much smaller, a much simpler system than C. elegans. And yet she keeps running against these roadblocks where if you don't have neuromodulators, now models work okay. And as soon as you add them, then everything fails. And so I don't think it is for lack of trying. I think that figuring out how C. elegans works is truly at the moment, in the ways, with the tools we have in science at the moment, a really difficult problem. And it might be that what we need is the ability to say, not just have the activity of each neuron, but maybe have it be really finely resolved. It is, based on what we know about C. elegans, entirely possible that it's not enough to have the activity of each of them, but you need it every 10 micrometers or something. And in this case, well, it's a crazy difficult problem.
Well, I mean, it seems like this focus on C. elegans. I'm not an expert on C. elegans at all, but when I have asked C. elegans people these types of questions, the response that I usually get is that there is a tremendous amount that is not known about the individual synapses and their electrophysiology and the individual neuronal types and their electrophysiology. And so there is from the connectome, there is dozens of pathways that a particular behavior could be channeled through, and throughout those pathways, you've got neurons and synapses that are not well understood, even from a neurotransmitter and receptor level. If I'm wrong about that, I might be 10 years out of date, and all of the sequencing and mRNA studies, or who knows what, has.
No, but you're right.
Okay, so in that case, it's a very simple answer. There are some very crucial. If we had an electrical circuit and we didn't know the values of the resistors, and we didn't know what the base and collector and emitter was for the transistors, then we would be lost. And if we even had a system with whatever the number that the Eve Mader's system has, I don't know how many neurons that has, but that could overwhelm if we didn't know the individual component parts. So I think that's the answer.
Yeah, but it's interesting how hard it is to identify it. Like in the ganglion, you can record from lots of neurons at the same time, potentially the bulk of or the main types of neurons. And yet, identifying it based on physiology is very difficult.
So, for instance, in C. elegans, not C. elegans, in Drosophila, the people around here are doing, there's lots of different neural types, and they're doing mRNA-seq on each of those neuronal types to try to see what the receptor types and what the neurotransmitter types are, in concert with trying to understand the system. I guess what I'm saying is it's early days in some of these invertebrate systems, and I would swing it back to comparison with visual cortex, because if I look at the literature in visual cortex and say, okay, we've got we've got a pyramidal cell that has a let's say we have a layer 4 pyramidal cell that is that's getting input from thalamic inputs, and it has a certain receptive field, and we have characterized the excitatory inputs, what those synapses, how they behave, how the neuron generally integrates that information. And then we ask something like, can we predict the receptive field properties of that neuron? And I point to experiments where they record from dendritic spine inputs and are able to show that those dendritic spine inputs that have certain receptive field properties are integrated into this particular neuron to give it a receptive field property that would be expected. It seems like it's a more an easier to understand system, or maybe not easier to understand, but people have been looking at it for longer and making a certain amount of progress on it. So I'm just, let me play a devil's advocate. I would say that unlike what Konrad said earlier, I will play the devil's advocate and say that C. elegans is hard because we don't know very much. Drosophila is hard because we don't know that much. But there are aspects of systems like visual cortex where the main, and this again, this is the devil's advocate, so I'm expecting Konrad to point out specific examples where this fails, because I know that there's general examples. You can always throw water on something and say, look, it could be that there's a protein that we have never heard of that does everything. But if that was the case, then we wouldn't be able to do these experiments that predict receptive field properties of visual neurons.
So I want to point out something about tuning curves. So in receptive fields. So the idea that you can describe a neuron based on basically which stimuli strongly drives the neuron and which other stimuli don't, it's not a given that that's even possible. And let me back up what I mean with that. So what people do in vision physiology is they show all kinds of stimuli, usually flickering plaids or something where you see like bright spots that move or bright spots that appear at random places. And then they fit a linear function to that. And they call that a tuning curve. A tuning curve by itself doesn't produce any real insight into how a circuit works. Namely, what we want, if we really want to have insight, we will demand that it generalizes. So what that means is, I don't want that I just take some class of stimuli and I fit something to it. I want to be able to predict what would happen if I would show you something new that isn't already in the training set. And it turns out that in the cases where people map tuning curves, in general, most physiologists never even go and do a real generalization study. And when they do, it's extremely rare. And when they do, it's typically a failure largely. So what I mean with that is if you probe, say, the visual system with like some class of stimuli, even if those are natural stimuli, and then you test it while the animal, say, hunts for food, you will get very different results. And the reason is because you made the logical mistake before that what visual cortex neurons do is calculate a local summary of what's in a stimulus. But that's not true. And we know from, say, Matteo Carandini's work that in visual cortex we find all kinds of activity related to movement. We know from Regenka Sear's work that we find all kinds of activities related to expectations. So in a way, the reason why it looks like we are succeeding in many areas of systems neuroscience is because we don't try to answer the hard questions. If you want to do whole brain emulation, it's not enough that you can fit me choosing like a bigger reward when the bigger reward is right and the other reward if the bigger reward is left. Not like those are the kinds of experiments we do. What you want to do is that you take something, you fit the model that describes what's happening within the brain, and then from that you can generalize to like everything. That's the whole point. And we're nowhere near that as a field at the moment.
But taking that specific example, I'm not sure if that's a fair example, because somebody would say, of course, there are other inputs that are not being that we're not seeing, but that doesn't mean that given the constraints of the study. So, what I'm trying to say is that how does science work, right? So science works by saying, here is a model. I can't test every aspect of the model, but I am going to use my best experiments to be able to test whether this model stands up so far. And so when somebody does an experiment that is very hard to do in the first place on visual cortex, and tries to get a quote receptive field, obviously they're not going to get all the inputs to that. Obviously, they're not going to get modulatory influences that occur at different times of the day and under different behavioral circumstances. But they're testing the model of, does connectivity predict function well enough to support the idea that the inputs to a pyramidal cell are being integrated, multiplied by the synaptic weights, roughly. And those experiments could have failed completely. They could have been, there is no relationship between the feed-forward connectivity or the sizes of synapses and the receptive field that you measure under, of course, the receptive field stimuli that you get. They could have failed, but they didn't, and so therefore it seems like we're making progress. Whereas I'm not familiar enough with C. elegans or Drosophila in particular instances. I mean, there are particular instances where it is widely the same as the visual cortex, but it seems like there is a margin of success that is occurring in these mammalian systems.
But let me take the opposite side of that, and keep in mind, everyone, I tend to in arguments play both sides. So, but the radical version of the other side is that the definition of success has been systematically wrong in that area. So if you say an experiment could have failed, let's go to sketch super simple experiment. I turn on the light, I see what's going on in some cells. I turn it off, I see what's going on, and I find that there's some cells that are, say, more active when I turn on the light, and other cells that are more active when I turn off the light. And indeed, this is real, and it succeeded at some level. Now, like if I give you a new animal, pretty much anyone, you will find some cells that relate to like the light going on, and some other cells that relate to the light going off. However, that doesn't mean that what the cells do is tell you about on versus off. In fact, it's like arguably the equivalent of stamp collecting. You know, like basically there is, if the brain is complicated, as we argue in the what does it mean to understand a neural network paper, then there is no, basically the model space is screwed up. So I can describe any number of phenomena and I get it significant, and if you repeat the experiment, you get the same results. So in that sense, it comes with that notion of success, but the results don't need to mean anything. It could be that it doesn't at all describe what these cells do when you and me talk or something. So the question is like what we mean by success, and I want to like zoom out of that for a second. So at some level, if you're into whole brain emulation, there are two very different ways of doing it. Either you can hope that there is a simple model of the brain that we can actually come up with, and that's possible. Now, in that case, the idea is right. I hypothesize a model, you hypothesize a model, our models battle it out based on data, eventually we converge to something. But that is only possible if it's simple. If it's fundamentally complicated, the only approach that we can have is basically hope that we find ways of basically getting so much data out of the system that even if there is no small model, we can still simulate it and it works just fine. Now, and where we are, I don't know. It turns out that physics is clearly on the simple model thing. Now, like the models they have in physics are so short that a human can like guess it, and that's how physics evolved in a way over time. Now, like you solve someone else's equations, you were like, oh, hey, I can actually make this more general or like simplify it and things like that. The question is neuroscience at all in the domain where you can meaningfully hypothesize, oh, here's a model of how it works? Or is it possible that because we learn in the world, such a model can never exist? In which case it means a lot about how we want to understand brains. It doesn't necessarily mean a lot about how we want to simulate brains.
So if I can just jump in for a second, I actually think that there is a ground where the two of you see eye to eye, because when you think about the two examples that you gave, Ken was talking about predicting receptive fields by looking at details, fundamental details of the neurons involved. And so that's more like the physics question, where you're trying to understand fundamental mechanisms, and from those together you're making a prediction. And Konrad, you were talking about an example where you're looking at a system and you are imbuing some kind of meaning to the fundamental components by doing a correlational study, which is really a different way of going about it. And it's almost like you're contrasting, what you basically said in your paper that neuroscience is doing wrong right now. This correlational approach is kind of the wrong approach, saying these are the neurons that do X. Whereas the opposite approach, where you're studying the fundamental components of a system, and then you are trying to make predictive models based on that. That is the model. That is the approach that is, heavy on data and fundamental components, which are, you've got this physics approach of describing how those work. So I'm not actually sure that you're disagreeing about the fundamentals of how people should go about it. It's just that maybe you are, you may not agree on what they can accomplish at this time. So that kind of brings us back to the C. elegans versus V1 question. And I see what Ken is saying about, well, maybe we've studied V1 more and we understand more about these fundamental components, so we can make more predictions based on that. It seems to me, though, that if we have the methods to do those studies in V1, we should also have the methods to do this in C. elegans and to do this in Drosophila. So if there was an interest to carry out the necessary studies to have the knowledge of the fundamental components and to then make predictive systems based on that, that should be possible. So I guess I'm still wondering why, after all these years, that hasn't been done.
So at some level, if we can't do C. elegans, it will be very difficult to do anything else. And I agree with you that really going for C. elegans, and like maybe succeeding, maybe failing, that at some level relates to Ken's debate with me. But really trying it is something that one should definitely do.
Mm-hmm. So I feel like C. elegans, V1 is always more complicated than C. elegans. Right? So I don't think we know enough about V1 at all. Right? So, but back to what Konrad said, I think I largely agree with with Konrad's comment. So the way I see it, we are talking about two different kinds of model, right? What Konrad really wants is this, I would call that ultimate generative model, right? All the small components and all the different combination of input output. So you can predict for arbitrary output, for arbitrary input, right? But on the other hand, what we have now is largely discriminative model. For example, like the input-output model I've been working for so many years, it's nothing else, but it's a conditional model, right? Given input, the probability of the output. So that's only one aspect of the generative model, right? In some sense, it's not a real model, right? And it's a way of organizing data. It's almost like the data itself, right? So I do see, obviously, there's a value of doing this kind of model, because whenever you have predictive power, you got some knowledge about the system. But I agree with Konrad. This is far from the ultimate generative model that allows you to explain and predict everything you have to predict.
Yeah, I think we all see eye to eye here. Not like Ken, would you disagree?
I think we all see eye to eye, but I'm trying to find the kernel of disagreement, if you will. I mean, I think that the question here is, and I want to double check this with Randal, but the question that I see is there might be. Obviously, we can't do emulations today of any significant complexity. That's obvious. There are two possible reasons for that. One is that we have a fundamental lack of parameters nailed down. And the other is that it is a really difficult problem, even if we nail down all those parameters. And so those are the two possibilities. And there's some pieces of research that are held up as saying of supporting one versus the other. And I'm trying to understand, are those researches actually really saying something deep that that says, look, we'll never be able to understand, if we had the entire connectome, let's push it back to the retina, okay? If we had the entire connectome of the retina, we wouldn't be able to predict a single output of a retinal ganglion cell, given a visual stimulus, because it is, even if we had the connectivity and all the ion channels, because it is such a complex system that it the level of predictability would be that if you plugged that simulation directly into somebody's optic nerve, that they would see the world in the way that a real retina would. And I look at the neuroscience literature, and it seems to me like we've made great progress on the retina, so that we can actually say, look, if it's in this layer and it's of this morphology, it's a good chance that it has this general type of receptive field property. So I see the weight of the evidence going toward the idea that it is a lack of understanding of parameters, and it's not a something more fundamental. But aside from that opinion, I think that's the real question that's being hammered out here. Is that correct, Randal?
Yeah, I think that that does describe it correctly. By the way, you mentioned that there are two possibilities. Now, there's, of course, the possibility that both hypotheses are correct to some degree, right?
Yeah.
That we are lacking in both.
I totally say yes to both.
Well, it's certainly the case that we're lacking parameters, but yeah. So nobody is doubting that. The question is, does the preponderance of evidence, or even forget preponderance of evidence, where are the experiments that really address the question of is this a lack of parameters, or is this something more fundamental? And I would argue that there and this is why I bring up V1. That there was always this discussion of, can you really understand the simple receptive field properties of visual neurons without taking into account all of the, by taking into account the feed-forward aspects, without taking into account all of the recurrent aspects and the modulatory aspects, et cetera, and given that there is a lot of experiments that seem to that seem to get quite far along the line of explaining receptive field properties based upon feed-forward connectivity. It seems to argue that the rest of the complexity is really we haven't hit that wall that you were talking about with Eve Marder. And so maybe this wall of complexity doesn't come up in some systems, it comes up in other systems. I don't know. That's my take.
But hold on. Let's try and make those numbers a little more meaningful. Now, like, you can look at Surya Ganguly, say, predictive models for the retina, for both retina and various people's models for primary visual cortex, and you'll be shocked. Now you get like R squared is 0.5 or something, which is really amazing. It sounds as if we describe half of that. Now, let me give you a system where you'll see how like insanely misleading that can be. Let's take your computer. Let's assume, and I don't know what the current voltages are, but let's assume for the moment if a transistor is on, the voltage is 3 volts. If it's off, voltage is 0 volts. Now, if your computer is off, which I hope it's 16 hours a day so that you get to enjoy life, then during that interval, the voltage is exactly 0. In the remaining 8 hours, it will be half of the time on, half of the time off. And everything that makes a computer is the fact, like how it switches. But if you have the simple model, during the day, it's 1.5 volts, and at night, it's 0. That model describes the bulk of the variance. But the model is entirely misleadingly, harebrainedly wrong. It's like it carries nothing of what makes the computer a computer. And that is the difficulty. Like, how do we know what these numbers mean? Now, let's say we can predict half of the spikes in a way, like we can predict half of the variance if we've been it by some time. But like, what does that mean? How deeply does it mean that we actually understand something real about the system versus maybe it's just like, let's take the extreme case. We know in primary visual cortex there's neurons that are very active if the stimulus is horizontal. They are not very active if the stimulus is vertical. Now that could be because the neuron is representing verticalness, but it could also be that the neuron does something incredibly complicated. It's just turned off. There's nothing horizontal in the area because it's like looking for things that have like some horizontal aspect. We would never know which of them it is. In one case, we discovered everything that's to discover about the neurons, or the bulk of it, because it's a detector for horizontalness. In the other case, we just discovered under which circumstances it does something. Now, where on this continuum we are, where we've either like basically learned only nonsense, or like only stuff that is real but is of no relevance to computation, and towards we understand almost all of it, we don't know.
But I mean, the papers that I'm reading aren't so blatantly taking the simple stuff. For example, you're talking about neural code. Let's say, we've got papers that can decode place cells in the hippocampus that can read out the position of the animal in the room, that can actually read out what they're thinking about when they're sleeping, and reward that, and then have that cause a behavioral change in the animal the next day, so that the animal thinks that, or behaves like it thinks that there's a reward in that particular area of the brain. So to me, that's like, here is an experiment that isn't just saying is the light on or off. Here is an experiment that's saying, do we understand the neural code well enough to decode the position of the animal from its neural representation?
But decoding doesn't add any information to it. Like decoding, let's go back to the computer thing. Yes, I can totally well, based on the average voltage on your transistors, decode if it's day or night. But it still is the wrong thing.
Well, if you could decode if you could decode my GPS position from the transistors in my phone, that would say something of your understanding, though, right?
But let's make a place cell model that suffers from the same problem. Imagine these cells aren't place cells whose role it is to represent which place we're at. But imagine, I'm not believing that model, but I'm saying we can't even rule out those simple models. The things that we currently call place cells are exclusively models that deal with thinking about the things that locally are where they are. So instead of that being like a I'm home place cell, it's like a do I like that image of the wall cell? We would never know, because it's still that cell would only be active when we're at home, because that image isn't there if we're not at home. No, but you see that basically it's not only that we have problems in neuroscience, it's we don't know what the nature of our problems is, and based on how we approach neuroscience, we cannot know how much we know about neuroscience.
But the decoding, if the way that we're doing the decoding, so you were saying, how well does a model predict things, and you could get an R of 0.5, and it was a essentially useless model. Any decoder that we are using to look at those place cells has built into it certain assumptions about what the neural code is. And if we had no idea what that neural code was, if we were so off as you seem to be implying, then it would seem like every one of these experiments would simply fail.
So, but what does it mean? The failure mode would just be that my transistors during the day would be the same average voltage as my transistors over the night.
Yeah,
so basically, that failure mode is not the right one. So basically, the experiments have been designed so as long as variance exists, they come out as success.
Okay, but ultimately, this does still, to me, sound like an argument about scale. Because these same experiments that you're talking about that fail because we're doing it at such a high level where you're looking at the entire computer and the voltage, and you're saying something about its voltage at night, its voltage in the daytime, and that prediction that becomes basically meaningless. If you do the same thing at the scale of the individual chips, where you're actually registering from each one of the little pins that comes out of it, and you can look at all the zeros and ones described as voltages of 0 or 3 volt, then you can actually begin to map what those chips are doing. I mean, we know this because they are digital chips, and that kind of reverse engineering is possible. So we know that this complaint, it breaks down at some point. At some point, it actually becomes possible to describe what the system is doing in the example of the microprocessor when you take it to the right scale and you're doing the same thing. So it sounds like more of an argument about whether we're looking at the right scale here when we're doing these experiments.
Yeah, it's the proof into the opposite direction is always doable. If someone says this is a cell that cares about color, or a brain area that cares about color, yes, someone can come, and it happens in neuroscience over and over and over again. If someone says, no, this isn't only color, this is also motion, or this is not only motion and color, but it's also texture. And
of course, you put whatever input into the cell that you want, right? The cell is just a processor.
Yeah, right, but it's difficult to go the reverse direction. I can say, here I find something that people haven't found before, but with the way neurons operate, it's essentially impossible to say we understand that now, because if you look at spikes, let's say of the neurons that we best understand in the whole brain, we still find that there's huge variance on when exactly the neurons spike, under which circumstances, how it generalizes. Like, there are all those things that we clearly don't understand at the moment. And yeah, maybe it's just an issue of amount, but basically, if you have an R squared that says 0.5, the idea that you understood half of how it works might be massively misleading.
True. Yeah. By the way, Dong Song has actually explicitly looked at how to compare different, patterns of spiking output and to say whether they fall into the category of a correct output and false output, even if you can't predict the exact spiking. This is part of what was necessary for the building of the MIMO model for the hippocampus. And so I don't know, Dong Song, if you wanted to add something to that, but if you have any nuanced view of what it means to understand or predict the probable output for a system, even if we can't predict the exact spiking.
So what we did, we had two kind of model, right? One, what we call MIMO model. It's the input-output spike model. The other model is what I call memory decoding model. It's looking at the spiking behavior, try to decode what kind of image the patient was seeing, right? But those two models, they are still a kind of a discriminative model, right? Input-output model. It's given the input, what's the probability of having another output. So it's very useful, right? It's useful for building the prosthesis. But obviously, this is only deal with a very small subset of the total population, and this is only work on the input-output relation. It's not what I would call the ultimate generative model to explain how the hippocampus work yet, right? It's far from that. So, but I do believe these are not overfitting model. We capture some important property of the hippocampal circuit without even understanding how the neurons they are connected, without understanding how this correlation is formed. But those correlations will be useful, and help us to get some deeper insight to how the hippocampus work. Because if you have your electrode in a neighboring region, it's a non-memory related region, you don't see this kind of correlation between the image category to the spiking activity. So that tells us something, right? Tell us at least hippocampus during memory formation, it generated this high level of correlation between the images and the spiking activity, right? So I believe we got a deeper understanding to the hippocampal function by doing this, although we are still very far away from having a complete generative model to explain how this correlation is formed, right, by all the components of the circuit. Hello? Is somebody?
Yeah, sorry. I was just allowing other people to jump in if they had a comment.
We do have one question from the audience from Abolfazl Alipour. Isn't prediction a step towards explanation? E.g., in Hodgkin-Huxley model, no one knew why we have an inactivation gate at first. It was just helpful for fitting the model output to actual data. Later on, we found that there is actually a protein acting as an inactivation gate.
Yeah, I can take the question. Like, yes, predictive models are typically an important part of getting towards a deeper understanding. Like in physics, we had lots of predictive models for phenomena, and now it feels that we understand them more deeply. So by all means, and I should say, I'm a big fan of models that describe variance in neuroscience. I'm just always promoting that we're realistic about where exactly we are at the moment.
So
I still feel like this is a scale issue, by the way, because, again, the example that Abolfazl gave, this is about fundamental mechanisms. And there, the predictive power of that model gives you something. It gives you a bit of an explanation of the mechanism that's there. And then you can put all of that together and you can create larger models out of it. Whereas, of course, the caution that Konrad, that I think you're throwing in there is, if we do the same sort of prediction at the level of an entire region and we say, oh, these cells are all telling us where we are, they're place cells, that then it becomes, something that could be sending us in the wrong direction.
Right.
I really still think this is about finding the right scale. So part of the question here is, do we today know the scale that we should be looking at? If we wanted to make an emulation of C. elegans or of Drosophila or of V1, do we know where we should be using our predictive models, building our mechanistic models from those predictions that we've accomplished with our experiments and studies?
Mm-hmm.
Or are we still too far off? Do we need to go build models at the molecular level and below? Is that what we should be doing?
Randal, I'd like to add on that. So in addition to scale, which is obviously important, another factor is what kind of signal you look at, right? What kind of signals you pick as your input-output data, right? That's basically, that constrains what kind of question you can answer. So for example, if you want to predict some brain state by using all the spikes in all the neurons recorded, I think that's a very promising approach for understanding the brain. But if you choose the input signal as EEG, right? As EEG, you try to predict from EEG how the brain works and what brain states the person's in. I feel like you probably pick the wrong signal, right? Now, that signal is not a real intrinsic signal of how the brain works. But
so I've got to run. Oh,
go ahead.
Sorry, I was just going to say, I've got to run. But my answer to the question, do we know at which scale we want to be, would be no. But like, I would love to brainstorm more about how we would find out what the right level is.
Okay, that's fabulous. Thank you, Konrad. I understand that you have to go. Your contribution was really very useful, and I also think that this conversation is definitely going in the right direction because we're talking about very basic things that neuroscientists sort of agree on in general, but then when it comes down to the details of what we should be doing, we probably disagree on a few things. So I think it's a very interesting conversation that will be continued. We'll continue here, but thank you very much, Konrad.
Great, thank you.
I would like to jump in, if I may, and talk about this scale issue.
Yeah.
Yeah. So there's this idea of causal emergence, and basically the idea says if you look at the system at different scales and different levels of zooming in and out in the system, and you study the causal interaction between these units at different scales, you will find a scale in which the causal interaction is maximal. So, for example, if you look at the logical gates in a circuit, you could find some sort of index for this causal interaction between these units. But if you go one level higher at the level of a copy gate, you will see some sort of different number for this causal interaction, and that's bigger because at the level of copy gates, this causal interaction is higher and more important for this system as a whole. And I think this would be some candidate for us to look and study what is the level that we should be looking at brains. Is it the level of ionic channels? Is it the level of neurons? Or is it the level of brain regions or circuits? And so far and so forth.
Yeah, that's a good methodological idea.
Right.
Yeah.
But there's always this issue about how to measure causality, right? In my experience, whenever we try to build a causal system, build a causal model, we still largely rely on correlation-based method, right? When you try to, based on the observed signal, you try to build a causal model, we define causality largely based on the Granger causality, which is still correlation-based. It's how much predictive power the input can provide to predicting the output. So it might not be the real biological causal relationship.
No, that's true, but I think it's good to at least have some ideas of how to go about picking what matters and what doesn't, which is what, to me, Konrad's Konrad's skepticism and careful analysis I really appreciate. But I mean, he often doesn't provide specific ideas for how you can limit the uncertainty. Right. So he'll say something like, we can't know anything, but he doesn't say what you should do to know something. So I think in that sense, this is very useful. Yeah.
The example you give, I think, is very good. So if you go to a computer, if you can go to the level of every logic gate, right, and you identify what kind of gate that is, and if you can replicate every gate of the computer, you can fully replicate the computer, right? That's a great example.
Oh, I disagree. You have to know where all of the impurities in the silicon of each and every gate is.
Thank you, Ken, for providing perspective. But yes, see, this is actually the reason why I keep mentioning computer emulations and emulations of computer circuits. And we've done this before in a few other workshops where I explicitly presented how do people try to reverse engineer circuits and that sort of thing, because I want things to be grounded in some sense of reality of, okay, this is an example of a system where we really do apply reverse engineering, and what does it take, and what is actually possible and what isn't possible, so that we don't, have to worry about the infinite amount of uncertainty that there is if we just philosophically think about all the things that we don't know. You've got this circuit system, and you can analyze it in a certain way. And so then I ask, why can't we do the same thing for a neural system? Let's say Elegans. If we do the same thing there, can't we get to the same kind of ability to reverse engineer what it does and to emulate it?
Mm-hmm. Yeah, by the way, I personally believe it's totally possible to fully emulate C. elegans. I'm not saying I can do it, but I totally believe somebody will do it pretty soon.
Is there a reason why? This is just totally hypothetical, but if you believe that it is possible to do it, is there a reason why you would personally not choose to do that? Do you think that C. elegans is not the right system to demonstrate?
Yeah, I think that's a perfect system to demonstrate this. It just, I need to think about it.
So if we chose a simpler system, and how about a neuromuscular junction? And we wanted to fully capture the neuromuscular junction's input-output relationship and simulate it. It appears to me that would be trivial, yes? Does everybody agree that a neuromuscular junction, even though it is fantastically complicated, fantastically complicated at the molecular level, that is too trivial to even contemplate quote emulating?
Right.
Okay, so I would say it's been done.
Okay, so I'm not sure if I'm not sure how many people would agree. It would be an interesting question to ask at a neuroscience conference. Because you could say, do we understand a neuromuscular junction? And probably half the people would say, no, of course we don't understand a neuromuscular junction, because we don't understand this particular biochemical pathway that is making sure that it reaches homeostasis under these circumstances. And from our point of view, we'd say, look, that's not what we're interested in. We're interested in, could we rip out a neuromuscular junction and put in an artificial copy of it and have it essentially do the same thing? Right. And the answer is, to us, obviously, yes. And I feel like we're at the same level with the retina. I could be wrong, but when I ask people, do we understand the retina enough to emulate it, I get answers that are in the negative. Whereas at the same time, there is a tremendous amount of research going into creating artificial retinal implants.
A slight issue with that is, say, the retina is another one of these examples of a potential prosthesis that's at the outside, at the rim of the system. It's external to us. It's kind of just an input. It's like a cochlear implant, right? So even if we say we understand quite a lot about the retina, and therefore the way that we emulate it, the way we build a prosthesis would be more similar to the neurobiology than, say, a cochlear implant, you still have the situation where pointing out that patients can use a retinal implant because their brain gets used to the signals and learns how to work with them isn't the same thing as actually reverse engineering the retina. And that's probably where you can get some of those complaints from, I think.
Yeah, so
I didn't need to stop the conversation there.
So back to I like the neuromuscular junction example. So I completely agree. In terms of computational function, we fully understand how the neuromuscular junction works, right? So it's a trivial modeling goal. But on the other hand, if you want to use protein, you use ionic channels, you use all the signal pathways, right, to fully replicate real biological neuromuscular junction, right? We are not even close. Right? But when we try to, we really need to define what emulation means. So we need to find the right language, find some computational function, right? Because otherwise, people will always argue your model doesn't include a certain kind of ionic channel, which is critical for which is really important for the neural circuit. So your model failed, right? So we need to define the goal as some kind of or certain behavior.
That's definitely true, I think, in the general case, that we need to understand what the goal is in order to be able to make any kinds of statements about what kind of emulation is possible versus not possible with the amount of knowledge that we've got. So the goal is important there. But, when we're talking about things like reverse engineering C. elegans or the retina, I think that enough of what this goal or success criterion is sort of generally accepted. Sort of, say, from C. elegans, you would like to see the same kind of behavior. You would like to see a nematode behavior. Mm-hmm. And in the case of the retina, if it were a really good reverse engineering of the retina, then you would probably want to see very similar signals heading to the brain as what you would find in a biological retina. And that's, of course, where then you start to argue about things like scale in terms of what is the resolution you're trying to achieve in your retina, and do the signals have to look exactly like the spike trains that you would see in a retina. But okay, I agree. I certainly agree that defining terminology is important. Defining success criteria and goals is important, because as long as we're talking about who knows what, everything is up in the air. It's the same problem when people come up with, discussing theories of consciousness or something like that. If they don't agree on what they're talking about, then you get all sorts of different weird theories. But I think, sorry, yes, go ahead.
Well, I was going to say that I'd like to say that the proper level is the level of representations. So, if we were trying to talk about a computer, we want to talk about the representations of, let's say, numbers in registers, and it may not it doesn't really matter at some level what the gates are doing in the algorithmic logic unit if they're actually doing addition under this circumstance, right? So, there is a tremendous amount of neuroscience that is trying to get to the representational level, and there are a whole bunch of hypotheses. Obviously, one of the hypotheses that gets thrown around continually is that we have no idea how insanely complex representations can be given spiking neurons. It could be the timing of every single spike relative to every other single spike. And okay, it could be that, but that has certain predictions. And so you've got a class of experiments that try to decode representations, and you've got a class of experiments that try to, if you will, stimulate artificial representations. And the decoding process is by necessity simplifying. And so you could imagine if the complexity of the representation is too much, the decoding becomes impossible. And the stimulation mechanisms are even more crude, like optogenetic activation, and or even optogenetic, or the stuff that the hippocampal stimulation with just a few electrodes. These are really crude, and yet they work to some extent. And so it says something concrete about those, what representations are important. And I would make it more concrete here. I want to be maximally concrete so that I can get people pissed off. That it's possible that the crucial representations are the spiking rates of neurons, and it's the coalitions of particular sets of neurons, often referred to as cell assemblies or neuronal ensembles, that are really the key representational units of lots of parts of the brain. I'm not saying every part of the brain. Obviously, but in a lot of cortex, you can decode stuff based upon the assumption that what really matters is a subset of the neurons firing together, and that firing together is an increased spike rate, irrespective of the exact timing of the spikes, over about 100 or 200 milliseconds. That is the coding hypothesis of what the representations, the causal representations are.
Yeah, that's a very traditional hypothesis you're making there, which is fine. Of course, I understand what you mean by people are going to jump all over you with arguments to the contrary, because there are some specific examples where you might show that, for example, spike rate isn't really the right measurement. But just in general, it seems to me that there are ways, as Abolfazl pointed out, to determine at which point going to the next level of representation, something at a higher level of complexity, doesn't give you any added ability to do causal inference. So there is, you've got diminishing returns. And the only way that it could be a problem to try to determine where you've got that cutoff is if going to the next level is prohibitively complicated so that you cannot make that determination, that you cannot make such a predictive model so that you cannot determine whether you have gained something or haven't gained something. I haven't seen any real evidence that this is the case. So I think we can probably, draw those conclusions. We can probably say something about which level do we need to work at. And, this has a lot to do with the whole question of whether or not, there's a scale separability in the brain.
Right.
This is really what we're talking about right now. I haven't yet seen evidence that this is beyond our abilities. So I think that if we tackle a system like Drosophila or the retina or whatever we think is the smallest system, probably not the neuromuscular function, I think that we can maybe get some consensus going among scientists with the right kind of publication, I would hope.
Yeah.
Can I ask Dong? So in terms of the hippocampus, I understand that the timing of ensembles, I'm going to throw the word out, is much shorter in sharp wave ripples and things like that. But is the general principle of encoding being the representation being collections of neurons that fire together, even if it's 10 milliseconds or 20 milliseconds, or is that still an accepted principle? Is there good evidence against that? Is there good evidence for it?
Yeah, I think definitely. But in hippocampus, timing becomes more important than firing rate, because hippocampus has a very low firing rate, right? So if you calculate like a mean firing rate, it's not particularly meaningful. If you look at place field, when the neuron is in the non-place field location, the firing rate is extremely low. It could be almost silent, right? And when the animal moves into that place field, then the spike generates almost instantaneously, right? So in general, that principle is still true. And also, I would argue in hippocampus, timing is probably more important than firing rate.
And it's coincidence of firing. There's I'm sorry to go back to the subtleties of neuronal representation or the neural code, but I think it is relevant. So there are temporal synchrony models that say that are simple and first order, and that I don't think anybody would disagree with. That says, look, there's timing signals in the brain that cause a bunch of neurons to fire synchronously to pass to the next decoder, if you will, to pass the information most effectively. But then there are temporal synchrony models that say that simultaneously there are two sets of neurons that are in phase that are representing two different things simultaneously, essentially. And that this is that essentially the downstream neurons that are decoding would have to not decode based upon just that I got all these signals at the same time, and then therefore I'm going to fire, but that are looking at subtle effects between when did this spike come in from this cell versus when did this spike come in from this cell, and it seems to me like there is a level of complexity in that direction that you can start imagining that would make whole brain emulation much more difficult. But the question is, does what is the evidence? What is the preponderance of the evidence say it is? Is it one versus the other? Again, clearly there's certain systems that are exquisitely tuned, auditory systems that are exquisitely tuned to spike timing. But in general, if we understood what the neural code is in particular systems, that would simultaneously explain what the level is that we should emulate. Correct?
Yeah. So yeah, I have some evidence to support. I think what you just said, right? Downstream neuron is actually simultaneously firing bunch of neurons, right? It's actually sensitive to a spatial temporal patterns of the input spikes, meaning they have offset in time and they all in the upstream region. Look at the memory decoding model. The MIMO model. The MIMO model really try to model what kind of input temporal pattern is most effective of the driving this output neuron to fire action potential, right? So that's this kind of asynchronized pattern. Those neuron ensemble, they don't have to necessarily fire at the same time to form an ensemble. They can have certain patterns with certain delay, right? So that adds another dimension. It's a temporal dimension. That will make the input dimensionality much higher, right? But I don't think in theory that's like a forbidding factor for people to do that. I like to go back to Konrad's nonlinearity is difficult to solve, but nonlinearity can be solved, right? he's saying like you have a 1,000 inputs. 1,000 inputs is not that big. It's still a solvable problem. It will make it difficult, but theoretically, I don't think that us from make it completely impossible, I guess. Yeah. But on the other hand, there is certain issue with this approach. For example, when we do the memory decoding model, because we are or even the MIMO model, because we are limited by the amount of data we got, right? So when we try to, for example, to decode a certain memory category, we have to sort of divide data into this category versus not this category. Sort of averaged out across many different images belong to that category, right? So I really don't know whether this category is a real internal representation of the hippocampus. It's just something we almost arbitrarily choose to make the classification possible, right? So the resulting model will be this category, this kind of division of category sensitive, right? Mm-hmm. So it works for this almost arbitrary definition of the two classes of the spike patterns, right? So that's the danger of it, because we really don't know what the internal of the hippocampus is, right? The hippocampus is very unlikely to, classify or categorize all the natural images to those images, right? So, but there are some existing try to find the internal representation using a sort of combination of the supervised and unsupervised learning, right? And I see that as a pretty promising approach. You find what's the real category of the hippocampus is, right? One may argue those colors are kind of arbitrary. It's based on the wavelengths, in certain colors, and one may even argue those shapes are sort of arbitrary. Right? It's something we measure, right? And not the intrinsic representation or intrinsic categories of the hippocampus, right? So yeah, I think one of the difficult questions is to try to find out those most meaningful representation of the memory. Because for now, all the models are kind of arbitrary. So we have to average a lot of the signals together.
Yeah. So going back to sort of combining what you were mentioning with Ken's question, I get the impression that there's still a difference between saying, okay, in addition to there being ensembles of synchronously firing neurons that have meaning, saying that in the hippocampus there is evidence that a certain temporal pattern of firing has meaning, which is what you've shown, that's still manageable, which is a little different from, say, if, as Ken sort of pointed out, an alternative hypothesis, if the exact timing difference between this neuron and the next neuron and another neuron, if that all matters and leads to a completely different outcome, if that's part of the representation, that's, of course, a different level of complexity, because then you have the system that is, in a sense, totally analog, because the timing difference between the different neurons is then the analog component. Whereas if you're just talking about a sequence of neurons or partial ensembles that together form some spatio-temporal ensemble, in a sense, that is still something kind of digitizable, even if it's, spread out over time. I'm not exactly sure whether this is where we have to make the distinction between something that can or cannot be emulated. I wouldn't say that, but I think I kind of get where Ken's coming from in saying that is a different level of complexity. Am I saying that correctly, Ken?
I want to make so I don't read a lot of hippocampal papers. I should, but when I read these hippocampal papers, there is often a decoding of a sharp wave ripple. Am I using the right terminology? Correct me if I'm not, because you guys are hippocampal experts. So
that's correct. Yeah, you talk about decoding a sharp wave ripple versus the hippocampus being in the mode where it's sort of controlled by, say, alpha and gamma rhythms and all that. Yeah.
Okay. So and they can look and see that there is a trajectory, essentially, if they're decoding place cells, and they can say, okay, this is under this time window, I am decoding based upon firing rate. The numbers, and it might be a 10 millisecond window. I'm not sure. But this sequence of 7 time windows is I can decode that as a representation of the trajectory that the animal is taking, and I can say whether it's being played backwards or played forwards and stuff like that. And so to me, that is not the type of temporal structure that is scary. Precisely because we can have we've had some degree of success in decoding it. Yeah, that's all that I'm really saying.
Right. Okay. Yeah, and I think that's true. I mean, so that's exactly what you find when you look at the place cell literature. But what Dongsong was alluding to, and again, I shouldn't be speaking for Dongsong, is that you find in the hippocampal studies they've done that you can take, say, a sequence of these kinds of activities. And I don't know if Dongsong, if you've even looked at it that way, but if you were to, say, take 5 consecutive ensemble activities in one of these 7 plus or minus 2, you would just wrap them up. You could say, okay, this is one representation. This spatio-temporal activity is one representation of something meaningful. I actually don't know. I don't think we've ever talked about that, whether you've ever looked at these spatio-temporal representations that you published, that you wrote about, and compared them with this previous literature on what I guess what's working memory, really, involving medial temporal lobes and hippocampus. But maybe this is leading us astray from the actual discussion, which was really more about, where do we go from here? As in, if you can indeed explore the connectome and you can indeed explore activity in neurons, and if we just assume that in 20 years, gathering all of that data isn't really the biggest problem. So gathering activity data and gathering connectome data is something that is more of an engineering problem at that point and just keeps on getting better. Then what do you do? How do you build these models, and where do we potentially run into hurdles, or what would you do? How would you start with that? And then we talk about these very fundamental things about how should neuroscience be done? How do you understand a system? What can be emulated? And why haven't we emulated C. elegans yet? I think that's where we're getting at. And maybe we're getting a little bit into the weeds by talking too much about hippocampal representations, although I understand why you brought that up, Ken. So I'm just sort of trying to steer the conversation back a little bit to make sure that we're not losing sight of the overarching goal here.
We do have... go ahead.
Go ahead.
Okay. We do have one question from the audience, Jan Klauk. If C. elegans and other rather simple systems are computationally too complicated, then we should go straight to the human and not waste our time/resources. What would be a hacker approach, i.e. perhaps science-based but not exactly clinical, we could do without asking an ethics committee that could potentially produce significant progress.
Oh, wow. That's going out on a limb. The reason I say that is because I think doing things that are not considered standard procedure, those are inviting extra scrutiny. So then you get looked at as, oh, why are you doing this weird stuff? I'm not sure that helps the cause as such of emulating brains. But anyway, I'm not really answering the question there, and I should leave that to some of our guests to try to answer.
Can you reinterpret the question? I'm not sure if I understood it. Randal, it seemed like you understood it.
Okay, so what in my opinion, what is his name? Jan Klauk was asking is, are there things that we can do to go get more quickly to the important answers by doing experiments or studies that perhaps avoid going through unnecessary steps like trying to emulate C. elegans or following other traditional protocols, maybe even avoiding, say, ethics boards and just doing whatever a neuro hacker would do to try to determine, to try to carry out studies that would help us move forward on whole brain emulation. So what could we do to accelerate the process? What are ways that we could do to short circuit some of the sort of tedious lengths to which neuroscientists go at present? And you know what? Even though I think that there is an aspect of this that is perhaps, that maybe is not taking into account how important it is for research in whole brain emulation to be careful to remain on the side where, we're just doing science and we're being taken seriously because we're doing science the right way. There is an aspect of this that kind of ties into Konrad's point about, well, maybe neuroscientists are spending a lot of resources and a lot of time doing studies that are going to be largely unimportant or useless to progress, as opposed to the kinds of studies that we should be doing. So, for instance, stop doing studies where you're doing correlational, giving results that are correlational based on, say, lesions or something like that, even though those are interesting and useful, and instead put more resources into studying fundamental mechanisms and gathering large data sets, be they connectomic or electrophysiology, and then learning how to interpret those, which is, I think, where Konrad agrees with what we are mostly talking about, that namely the science of emulation is actually really useful in a sense because it has a very clear goal, and we know that we should be looking at mechanisms and data. So anyway, that was just me reinterpreting, but yeah, the question was really about what can you do to short circuit this whole process.
Well, I mean, at one level, I'd say there's no way to short circuit this process if we're talking about whole brain emulation. I think I interpreted the question a little bit differently, so I will give my example of what I think hacking would be. So some of the stuff that people do in MRI. That say, gee, we can't record from every single neuron, and certainly not in a human brain. What if we look at the voxel level, the fMRI bold signal voxel level changes? Could we create a decoder that could make any sense of that whatsoever? And the answer is, holy crap, yes, you can. It's not very good, but you can essentially start to decode what a person is seeing or what a person is even imagining at a very crude level, based upon the several cubic millimeter voxel bold signals. That's it. To me, that's the kind of spirit of hacking that I was interpreting the question as, which is certainly has no ethical problems whatsoever because you're not opening up a brain.
No, this sounds more like what Jack Gallant is doing, what you're describing, really.
Exactly. That's exactly what I was referring to. So, I mean, I think from a whole brain emulation level, so let me say something a little bit more controversial. If you're talking about a whole brain emulation of the type that we're talking about, we're really talking about having to have, at a minimum, the connectome, the structural connectivity of the brain at the synaptic level. At a minimum, you have to have a set of side experiments that determine every single type of neuron that is part of the connectome, and its general electrophysiological and synaptic responses, and so that you can then create a simulation of that. It seems like that is the kind of the base level that you would have to get to, and considering that first the technology to create a connectome of anything larger than a Drosophila right now is many years away. And certainly the much harder thing is all the different types of neurons and their electrophysiological properties. Those are thousands of side experiments that are being done, but simply just have not been completed. This pushes any type of real emulation off many decades.
Thanks. I think that's a great response. I think I'd love to open up the floor to anyone else who is actually on this channel right now, just because I noticed that we have quite a few people who've joined us directly on the Meet session here, and many of whom we haven't heard from yet. Of course, maybe that's because you don't have anything specific you'd like to say or ask, but I definitely wanted to open the floor and say this is not just the Ken, Randal, Dongsong speak session. So, Michael, Cerullo, Todd, anyone who'd like to say something? If you have any comments on this, please go ahead before we move on.
I was going to say, I don't have a whole lot to add other than it was an interesting conversation, but I think some of the stuff Konrad was getting into really, it was beginning to touch on some, philosophy of science and some, epistemology issues that seem a lot deeper than neuroscience. So I thought that was interesting, but I'm going to track down some of his papers, but other than that, I'm just kind of an interested listener. So,
yeah, that's actually the thing that I simultaneously am happy about, but also worry about. I think it's great that Konrad is asking important questions. So, for instance, I think it's great that he is pointing out that perhaps neuroscience should be putting more resources into a specific type of research, and I think it's actually research that would help us a lot. But at the same time, there's the danger that if you're diving into, the philosophy or philosophical questions, which is basically always the question of what can we possibly ever know, given all the uncertainties that exist, there's always a danger of getting lost in that, which is why, I'd like to bring it back to, hey, here's how we can analyze a computer circuit. What if we did the same sort of thing for a brain? What would really happen? Anyway, just saying that's kind of where I get back to usually.
And just to what degree is he just generally skeptical about knowledge as opposed to just the brain, which would have been interesting to ask, but.
Yeah. Well, it does, his point does say something about neuroscience as a whole, because if you look at how much funding is going into neuroscience as a whole, then you can begin to wonder what would happen if that got redistributed in a way where, say, as he said, neuroscience should radically change. And if we did radically change neuroscience so that less funding is going into what we might call the standard approach, and instead going into massive neural data analysis and attempts to emulate pieces or whole brains, and the study of fundamental mechanisms. As he points out, you want to understand the learning mechanisms that are underneath, and then you want to make predictive models. You want to test those. You don't want to necessarily try to do all these studies where you're saying, okay, these are the Jennifer Aniston neurons. That could have a huge impact. And so I think it's a meaningful discussion, just in the sense that whether you choose one way of doing neuroscience or another and where all the funding goes, it could make a huge difference to where we are 10 years from now. I don't think that we necessarily have the clout to impact that right here, but it is a discussion that should be had in a larger sense. I am kind of curious now, for the people who are here, who are still on the channel, I was wondering, looking back 10 years, because, I mean, we're looking at this roadmap that was created for whole brain emulation back in 2007, 2008. I'm wondering, are there really any new insights from the last decade that would significantly change how we imagine that we could emulate something like a Drosophila brain? Is there something we've learned in the last 10 years that really changes how we would have, say, written such a roadmap?
So if I could throw out a possible answer to that, I think the deep learning revolution has given us a different perspective on things, and this is echoing some stuff that Konrad was saying. The idea that that a deep neural network does its job fantastically well, and yet if you look at the individual neurons, they do not mean anything. I think that was something that in 2007, 2008 was not fully appreciated. It is viscerally clear that you can get a collection of neurons and a simple learning rule, a collection of simulated neurons and a simple learning rule to learn grandmaster level chess with no difficulty, to learn how to drive autonomous vehicles, to learn how to do translation between one language and another at a reasonably good level, all with the same set of what we would consider to be extremely simple neurons, which are just linear summation devices with a simple nonlinearity at the end and a simple learning algorithm. And so that is the tall pole that has changed over the last 10 years.
Yeah, I completely agree, actually. I was about to say the same thing, because I'm not talking about just 10 years ago, right? When I was a graduate student, I remember I took this class of pattern recognition, right? So at the time, people still think we don't know enough about single neuron, right? Because, you know, like if you try to recognize an object like a cup, it's so difficult, right? Because, you know, cup, you know, depending on the perspective you look at the cup, every image is different. But the visual system somehow figure out how to generate this very high level representation of the cup, can classify images from different perspectives with different color into this single class, which is a glass, right? It's a cup, right? So at the time, about 20 years ago, people didn't even think that's possible. But as Ken just said, right, given the advance in deep learning, people at least, that's a very good example showing even you don't have you only have very simple neuron model, like ReLU, right, like convolution, that type of thing. You can achieve function as complicated as that. So given this, if you think about single neuron is always more complicated than ReLU, right? ReLU activation, those simple ways. So there's no reason the real neuronal network cannot perform, right? So at least theoretically, that indicates this is completely possible, although we still miss some details. But the complexity we have, the complexity of the understanding to single neuron is already more than enough for achieving very high order cognitive function.
It's interesting that you both point that out because, you know, I guess a curmudgeon-y response of mine would be that there isn't really anything that has changed in our understanding of how to build neural networks since 10 years ago and now. The main reason why deep learning neural nets are better at what they do is because A, they have much bigger available data sets to plow through, and B, they have computers that are really fast and have GPUs and can crunch through all of that data to do all of that learning, and that's why they can afford to have deep layers. So I would say there isn't really any new understanding in the system, but yes, it shows us what those systems are capable of if you throw enough resources at them and enough data at them. And that's valid. That's important. Not sure. Yeah.
Well, I agree with what you're saying. I am trying to read through Terry Sosnowski's recent book on this history to get a better perspective of how neuroscience played into this. But the fact that these simple learning algorithms had such success have also pushed back into saying how could they be biologically implemented, and so there is a set of biologically realistic models of backpropagation or things like that that intervening. I think the two of those things combined together make a powerful statement. But even if I was just taking your statement at face value, that the only thing that has really changed is faster computing and better data sets for training, so be it. The argument that people were having in 2007 was we really have no basis to go on to say that you get human level type intelligence even in via learning in these quintessentially human tasks by a neural network model. So it might be something fundamentally different that's going on in the brain than these models that we're proposing. And now that argument is just dead. It might still
agree. That's actually the point I was trying to make, because if it's really about the data that's being put into it and the amount of processing power crunching that, then that also says something about what is holding us up when it comes to things like emulating C. elegans or emulating retina or emulating Drosophila. It could be that it is largely the data that we've managed to collect and the amount that we've been able to crunch through, more than it is some fundamental problem that we haven't yet figured out.
Right. So if you look at the artificial neural network, right, I think decades ago, mathematicians already proved that if you have a multi-layer neural network, you can fit arbitrary functions, right? Literally arbitrary function. It's a universal function approximator, right? So that actually means you can do all sorts of things that deep neural network can do theoretically. But still, that deep neural network, convolution network, is still a great demonstration to show that's actually possible, right? So for example, if mathematicians only prove mathematically that's possible, at the time, there must be people like Konrad Kording, right, saying, okay, we still don't know a lot of things about this network, right? It will never work. Although you mathematically prove it could work, but it will never work for this and that reasons. And the best way of shutting them up, right, is you demonstrate that you can actually do it using a real demonstration, right, to show like you can actually classify all the natural images.
I would like to play the role of devil's advocate in here and say, so there are some problems in deep neural networks that point out to the fundamental flaw in these networks sometimes. So for example, if you use adversarial attacks and, for example, you just change some pixel values in a picture of a cat, and your network recognizes this picture of the cat as a toaster with like 95% confidence that it's a toaster. So there are a whole bunch of different examples showing that these deep neural networks are extracting the wrong set of features, sometimes, not always, sometimes. And so critics of the AI are saying that maybe we're approximating the wrong set of functions, and there's still a long way between where we are and where we can have a real artificial intelligence that can do a task that are equivalent to human performance.
Yeah, like people like Gary Marcus. I think that's right.
Yes, Gary Marcus, Melanie Mitchell. Yeah.
Yeah, that's correct.
I agree with him almost entirely. I haven't read his new book, so maybe there's something in there that I wouldn't disagree with. But I think it's more subtle than that. I think it's that it is absolutely clear that the brain doesn't work solely as a deep neural network that is trained via backpropagation. That's clear. But the power of this deep neural network module seems to be taken advantage of in a much larger, more complex system. I would make the case that that's the correct way of looking at this. So these adversarial attacks, if we were just by the way, there's some examples that I've seen of adversarial attacks on the human visual system. So there's even a little bit of example that it might be supporting. But the idea is that given a larger system, these neural network modules are combined with more subtle learning effects to get around things like these adversarial attack weakness, and like what Gary Marcus often says is just common sense reasoning. You know, there's no in most of the neural network models there is no place for kind of symbolic reasoning, and it's, you know, there are some purists that would say we don't need any symbolic reasoning, but there's a whole bunch of evidence from cognitive neuroscience that we are doing symbolic reasoning, and so I don't see that as a proof that we're not doing deep neural networks on some areas of the brain. I see it as a demonstration that there is larger scale organizational pathways that we haven't fully understood that make use of these deep neural network modules. I'm not sure if that made sense. Sorry.
It does make sense to me. And, you know, when you say that it ties into things like reasoning, it ties into a whole bunch of stuff that we might just call context, that we grasp that the network that has been trained on a specific individual task may not have. And that's an unfair advantage, of course, that we have all that context and those context-specific ways of dealing with data.
Yeah, the question is, I guess the real question would be the stuff that we are obviously able to do that current deep neural networks are not able to do, or are fragile and fail in ways that a human being would never fail in. Does that point to a fundamentally different nature of computation at the neuronal and synaptic and molecular levels, or does it point to a difference in architecture that really is something that Google DeepMind is going to figure out in the next 10, 20 years without positing something other than you know, the kind of standard neurons that are populating these models? And I think it's very dangerous to. I think we don't do this, but I think there is a tendency in the more general neuroscience or even public to kind of separate what the brain does into two general areas. It's the simple stuff and the complex stuff, and the simple stuff the neural network can do, but the complex stuff is so complex that it requires new physics. Even I mean, there is just a vast number of people that would say, yes, we can model the visual system with relatively well with a deep neural network. Obviously, there's people in the neuroscience community that would disagree with that. But you know, there are even people that would buy that, no problem. You can model the visual system, you could emulate the visual system, but you could never emulate higher order thought, or you could never emulate consciousness. And you know, this is the thing that that I think a lot of people that look at the grand goal of whole brain emulation will probably look at all of these sub experiments, all of the arguments that we've been talking about today, and say that none of them are addressing the real question.
It's interesting that you say that. This is kind of what I was going to get to next. By the way, I think that, you know, yes, I understand what you're saying that a lot of people will do there, but I feel that very often the arguments would fall prey to Occam's razor. Basically, you know, is there really no way that if they were, say, the engineer of the human brain, they couldn't figure out a way to build a system that just used typical neural processes to carry out the thought that they're trying to produce? Well, you know, they haven't really demonstrated that, first of all. But in any case, so yeah, just to kind of take what you just said and try to take it to what I might call the last big question of this panel is, if, you know, we've been discussing whole brain emulation of lower systems. We've been discussing retina, we've been discussing visual system, we've been discussing Drosophila and C. elegans, and some of the basic questions that we could have about that. You know, what is the scale at which we should be looking at the data, and how should we, what do we do about predictive models and all that sort of thing, and nonlinearities. Now, if we set that aside for the moment and say, because these are all the questions about brain emulation in general, is there something when you go from Drosophila to human brain, other than just the fact that it's bigger, other concerns that would be raised, something that you would have to take into account as you're thinking, okay, I'm trying to do this emulation of Drosophila here because that's what maybe in 2040, based on all the data, we can try to do. Next step, human brain. Would there be any additional concerns, anything that would be raised, anything that's important there? Or is it basically just a big Drosophila brain?
I think it's a big Drosophila brain. That's my
good statement.
Yeah, and my one piece of evidence for that is I believe I read somewhere that we're evolutionarily related to Drosophila.
I'm sure that somewhere down the line that's true, since we all ended up coming from these single-celled organisms, right?
Yeah, I'm just joking.
Yeah. Okay, I guess there aren't too many comments on that. So is the statement simply, we are a large Drosophila brain? That's something we can use as the exit statement for this workshop?
I'll stop at the rat.
Oh yeah. Okay. So then let me ask this: if you had done the experiment for emulating a Drosophila brain, and now you had to move to emulating a rat brain, do you see anything different besides just scaling things up?
I don't think there is. Right. That's like
right.
That's a working hypothesis.
Okay. So a rat brain is just a big Drosophila brain, basically, as far as reverse engineering or emulation is concerned.
Let me argue this. I feel like I have to take the other side now. certainly there is a level of learning and flexibility in mammalian brains that does not exist in the hardwiring, mostly hardwiring of invertebrate brain. I don't see that as being a showstopper. In fact, to a certain extent, I see it as being, if anything, making things ever so slightly less difficult, slightly less difficult. The reason being that Drosophila or C. elegans can, over evolutionary time periods, so overwhelmingly build complexity into its circuitry because it just simply doesn't have enough time to learn stuff. Whereas a human baby is pretty useless right out of the womb and stays useless up to, or if we're going to talk about a mouse, even a mouse is pretty useless over a certain period of time. But there's general learning mechanisms that, and so there is a difference. I don't want it to be any implication that there isn't a difference between the two, but I don't think there's a fundamental difference in the sense that one uses neurons and one uses magic pixie dust.
Right. Okay.
I mean, I should point out that maybe I shouldn't point this out, but Christoph Koch has a new book coming out whose subtitle is Why Consciousness Cannot Be Computed, something to that effect. I have it on pre-order. It's very interesting. But this is a by any account fantastic neuroscientist that is really saying that there is some fundamental difference that cannot be captured via computation. So it might be the case that there simply aren't the people here to argue that side of the story.
Okay. Yeah, I'll just have to reserve judgment on that until I get a chance to read his book, because I really shouldn't be judging it without seeing it. But I have a visceral sort of reaction because I've seen some of the things that he's spoken about before or written about before. But yeah, that's for another time and a different topic.
I mean, I would be, as a suggestion, I think it would be interesting, and you probably have already done this, but to invite some people that really do have the exact opposite perspective than we have to make the arguments.
What is the exact opposite perspective of ours?
Well, I mean, from Christoph's point of view, and Giulio Tononi's point of view, I assume it would be that we are taking computation as the sole thing that the brain does, and that is our fundamental mistake. That we have bought hook, line, and sinker the idea that the only thing that the brain does that matters is A causes B causes C, and it doesn't matter what molecules are doing that. It doesn't matter what order they're happening in. You can make tremendous amounts of you can take a system that is physically consists of a million parts, and instead of having them physically laid out and interacting in real time, you can simulate them one at a time on a computer and store the results in registers. And as long as it is a similar result, then it's doing the same thing on the inside. That's what they fundamentally think we have, that we're in error.
I see what you mean.
Yeah.
But it is. I mean, they're taking on something big there because they're not really just taking us on, right? They're taking on an entire approach to science. The whole approach that, yes, mathematical description and reductionism makes sense is basically what they're taking on there.
Absolutely, but I want to try to to step outside the whole brain emulation bubble that we've got around ourselves. Okay, the number of neuroscientists that are viscerally disagreeing that that whole brain emulation, mind uploading. Okay, that mind uploading is impossible. That they're saying, look, mind uploading is simply impossible. There's a substantial fraction that think that, and so if they're thinking that, you can say, look, they haven't thought about it deeply enough. Fine, that might be the case, but it may also be the case that they've really got some good thought arguments that we need to hear.
Possibly, yeah.
Yeah, we should get them in here and have them make the case. And we
That's a really good point.
have a big target on us, so it should be very exciting for them to burst our bubble.
Yes, yeah, I agree. And you're right. We haven't really taken on that challenge. We haven't really invited them in and said, hey, okay, make your argument, please. And I'll admit that part of the reason is that I didn't really know how to ask that question, because it doesn't seem like, beyond just saying, oh, well, that's impossible, I haven't really seen a lot of them say, and I'd like to explain to you why. That part seems to be missing a lot of the time.
Well, and it gets to, with Christoph, it's accepting Tononi's integrated information theory. And it's actually a little bit subtle, what is exactly wrong with that. But I think when you really, really analyze it, I think, as Ken was saying, that what they're arguing just really goes way outside of just what we consider just mainstream science. I mean, it's not as far out as like Hameroff and looking at magic physics, but it's quite a step away.
So I think Christoph would, even if IIT, if integrated information theory was wrong, I think he would. He has a principled objection to the idea that that consciousness can be explained through representational modeling, and it is a principled objection based upon what I think might be an error, but on psychological experiments that basically say, look, there is a difference between, he would put it this way, I think, there is a difference between experiencing something and modeling that doing a self-model that you're experiencing.
I just wanted to add to this idea because, I'm a bit sympathetic to the Christoph Koch's idea, and the analogies that he has used, and they really resonated with what I thought is stuff like this. So he says you can simulate a black hole inside a supercomputer, but you do not expect the space-time around that supercomputer to bend, right? You can simulate the storm inside a supercomputer, but you do not expect to open the CPU racks and then supercomputer racks and then see drops of water. And I think that's his main argument. And in a paper that he and Tononi published a while ago, they have shown two different architectures of neural networks, one of them feed-forward, one of them with recurrent feedback connections, showing that they both perform the same type of computations and the results are similar. One of them, the one with a recurrent connection, has a big number for the phi, the integrated information measure, and the other one has a lower measured number for that. So according to that, they say, and also this idea that people can consciously perform and have some sort of behavior without being conscious, they kind of argue that, okay, you need to have something other than just input-output relationship. You need to have this causal interaction between the units inside the system to recreate the consciousness.
Yeah. By the way, just quickly pointing out that it's funny that you would say that his recurrent network has a higher phi, because isn't it according to Koch and Tononi that the simulation of the recurrent network would still have a very low phi because it's a simulation instead of an actual hardware implementation of a recurrent network? So there's that. I'm just saying that because, I like to kick at that particular claim of theirs, because they like to make this distinction where you can calculate phi only over the physical transistors. I mean, not even the transistors, the physical, connectivity of the system, rather than say some representational connectivity of the system. But this does, by the way, I don't know if Keith is still on here, but Keith, if you're here, you wrote specifically about this question of, is simulated fire, does it burn, or is simulated water wet, that kind of thing, right? This is the same argument as the black hole simulator doesn't change space-time around it, right? Are you here? Not sure if he's still on. Let me check that there.
Oh, I think he left. But that was his paper, though. Yeah.
That was indeed his paper. Yep. Too bad that he's gone now that we finally got to that. But we don't really need to discuss this in detail, because this could take another couple of hours. But I'm happy to, let everyone address it anyway.
But so all that I'm saying is that I think we have something similar or in common with them, and that is that we are interested in the same question. We might be on different sides, and this, I think, is what sparked this conversation, is what is the opposite side. So people like Christoph and Tononi and others, they might actually be interested. I throw out to get a debate going, because they are willing to be on the opposing side. And I personally, when I am interacting with people regarding whole brain emulation, regarding the idea of whether neuroscience should be trying to address the question of what how memories are encoded, so that you can see what methods might work for brain preservation, etc. It seems like all of the types of experiments that we're talking about don't make a dent in the argument. And so it might be useful to put some energy into actually addressing the big consciousness question, the big C.
The hard problem.
Yeah.
The hard problem.
Yeah.
Exactly.
I think you're right. I think that's where we're headed to. We're slowly carving away parts of the problem and discussing the pieces that we can do before we talk about consciousness, just so that we can do this the way neuroscience likes to do, which is to keep consciousness out of it until you can't. But I agree that this is the thing that has to be addressed at some point. It's the direct conversation about what matters and what doesn't matter. Yeah.
Well, this is where I actually disagree with Ken. I mean, Ken, I think you just don't believe in the hard problem at all. Christoph believes the hard problem is a scientific problem. I believe the hard problem is a metaphysical problem. I think that's the difference.
I would love to. We should talk more on this. I mean, we've talked some on this, but I am interested in talking more on this, and it would be great to have Christoph on this conversation as well.
Yeah, we definitely need to have some additional invitees for this kind of conversation. But just briefly, Michael, because I'm a little bit confused at the moment, when you say it's a metaphysical problem and you say Ken doesn't think it's a problem at all, what do you mean by the distinction between those two?
Well, that's a bit tricky to I guess sum up in a couple of sound bites, but
because I was agreeing with your disagreeing with me. So I think what I'll give Mike's last. I think what I'm saying is that if we understand the self-representational properties of the brain, that we will have understood consciousness, because we will see that the system itself is representing the fact that it is in an experiential state. Full stop, done.
Right. So the experience of being is basically a state or an output of a certain system that does a certain calculation. That's what you're saying.
Sure. Yes. So, well, it's more subtle than that. But in any case, what Christoph would be saying is, no, that's not good enough. But there's this other property that is purely physical that will that goes under the name of this mathematical phi, etc. That will really truly address where computation is, where consciousness is coming from. And what Michael is saying, correct me if I'm wrong, is he's taking David Chalmers' point of view of essentially looking at all of these proposed theories of consciousness and saying that doesn't even get to what I'm saying. Why does it feel like anything? You know, I don't, why does a representation or even a self-representation feel like anything? Why does interconnectivity, integrated information feel like anything? Why does microtubule quantum vibrations feel like anything? None of them really address the why does it feel like anything question. And that's a metaphysical question. Is that right, Mike?
Yep.
Well, shouldn't one start by trying to discuss or define or to study what it means to feel something in the first place? I mean, anything, whether it's something as ephemeral as being and experience, or if it's something much more direct, like feeling pain when you touch a hot surface. Shouldn't one start somewhere by trying to describe what it means to feel or experience something?
But it's something we all understand.
Well, and there's definitely a decent, literature out there talking specifically about, what we mean by experience. Both neuroscience, analytical philosophy, and certainly phenomenology. So, I mean, I think there's a lot out there about that question.
Okay, well, I can see that this is indeed a large topic, probably worthy of many, many different workshops. I think we're coming upon, what is it now? Is it 3.5 to 4 hours, something like that, Michael? So maybe it's time for us to wrap it up and say that we've collected a lot of useful input.
All right, absolutely, yes. Okay, so we have now reached the conclusion of our panel discussion. Thank you all very much. Dr. Koene, do you have any closing remarks?
I suppose not that many, except to say that it was interesting to hear that on some level, the different participants in this conversation seem to agree that there are things we can say based on neuroscientific study, and that perhaps neuroscientific study does have to focus on details such as what is the level at which you should make representations. But on the other side, when it comes to really just sort of more philosophical statements about what we can know, that's where we tend to disagree a little bit more or talk past one another from time to time. But I think there's room here for progress. So if we wanted to take something like Drosophila or C. elegans and decide on a good way to move forward, and we really ask this question, where do we go from here? I think that there is some consensus on what one should do, what sort of data you should collect, and what kind of model you should build, and which questions you should answer. So there's some real practical stuff that can be done here, and this is the sort of thing that should be pointed out as we rewrite this roadmap. Then, additionally, we've discovered that, yes, of course, we can't continue to avoid the big questions. At some point, even this roadmap on whole brain emulation needs to include some part about why we make a distinction here between emulating Drosophila and emulating human brains, because that leads to mind uploading, and therefore there's a whole question about experience and consciousness and all of that. That also does need to be addressed, and of course that was not part of the original roadmap because it's such a thorny issue. I think that's about all I'll do in terms of summarizing or concluding. In any case, I'm very happy to see what kind of conversation came out of this. We tried a new format by simply presenting a couple of interviews because we felt that doing that we could focus the discussion more on the things that we really wanted to discuss instead of having multiple presentations where the presenters would present whatever research they happened to be doing in the past. Instead, everyone's focused on the same questions because the interview brought out the same questions. And that worked fairly well. And as we go forward, I think we're going to do some more individual interviews that perhaps will be streamed as their own thing without a workshop around it. And then eventually the next workshop coming up. Before we leave, I wanted to quickly ask Ken, do you think this is an opportunity where you would like to say a word about your upcoming conference, or do you think that should be kept separate?
Well, I guess I should say something about the upcoming conference. Well, very briefly, Randal and I are putting on a conference at the Society for Neuroscience. It'll be addressing the question of how long-term memories are encoded and what experiments might show that they could be decoded from, let's say, a connectome. It's really targeted at neuroscientists that can dig in and fight about recent papers addressing that question.
Thanks. I think that was very clear. So thank you, everybody, and thank you, Mom.
Okay, thank you all very much. We have reached the conclusion of our 2019.
I just dropped off there. Carry on, Michael.
Okay, we have reached the conclusion of our 2019 summer workshop event, where we go from here. I'd like to thank all of our speakers, panel experts, and our audience, our volunteer staff, patrons, and donors. Thank you all very much for making this workshop possible. Once again, we are a 501c3 nonprofit organization operating with the sole mission of advancing research in whole brain emulation. We appreciate the many contributions that have allowed us to organize events such as these. If you'd like to become a monthly donor or to make a one-time donation, you can visit our carboncopies.org/donate and donate by PayPal, credit card, bank transfer, or cryptocurrency. You can also contribute by volunteering. We are always looking for those who have a technical background to help with our technical review and anyone who has experience in the nonprofit world, such as recruiting, event coordination, fundraising, marketing, and so on. You can go to carboncopies.org/openings to apply. You can also sign up to our newsletter or follow us on social media at the Carbon Copies Facebook group, www.facebook.com/groups/carboncopies, or on the Reddit page, www.reddit.com/r/carboncopies, and on our Twitter account, a Twitter handle @carboncopies.org. All audience members are invited to take our survey. Our survey is posted in the video description on the YouTube channel. Thank you all again very much. Thank you, audience members, thank you, speakers and panelists, and hopefully we will all see you again at our next event.
Thanks, everyone.
Thank you.